Hi students, welcome to Unacademy NEET English. I hope I'm perfectly audible and visible. Yes, I am. How are you guys? Hi. Hi students. I hope I'm perfectly audible and visible. I hope I'm perfectly audible and visible. Good evening. Welcome to Unacademy NEET English. So students, you people requested us to start the game of NEET 2.0 again, and here we are. And today, the chapter that we are going to discuss is Biomolecules. So today we are going to finish Biomolecules, right? You know that it is one of the most important chapters. From this chapter itself, minimum 4 questions can come, right? Minimum 4 questions can come, number one. And this chapter is very important to understand the nucleic acid part from the chapter molecular basis of inheritance. So I always advise this to my students that please read the biomolecule, then go for the genetics, then go for the biotechnology, right? Then go for the biotechnology. So this is what we are going to discuss today. So excited people? Yes? Do you? Yes? Are you excited? Are you excited? Good evening. So after so long, right, we'll have a long marathon session again. Are we going to complete enzymes in the session? Obviously, we have to complete the entire chapter, na? So enzymes, right? Even the carbohydrates, the proteins, lipids, nucleic acids, enzymes, we will cover everything. Okay. So before starting the chapter, let me tell you students, you know that we have started our Avengers fast track batch, and let me tell you students, we have already completed one chapter here in biology, right? The batch started on 16th of December, right? Particularly on Unacademy platform on 17th of December we had our first class, and we have finished one chapter. Okay, we have already we have finished one chapter, right bache? And Sunday, that is tomorrow, they are going to give test as well, right? So if you have joined the batch, very good. Otherwise, please click on this link, be the part of this batch. Okay, be the part of this batch. And here in this particular batch, let me tell you students, here in this particular batch, the content is amazing, the session planning is amazing, right bache? And trust me, you will complete your syllabus till March. Okay, till March 31st your syllabus will be discussed in detail, and along with that, every week you people are going to get test. Every week you people are going to get test. Okay, and here in this batch, the session plan is in such a way that from one chapter, like see, we have finished cell cycle, biomolecules, then molecular basis of inheritance. So the sequence is of the chapter here is going to be amazing, right? So without delay, be the part of this batch. And obviously on YouTube, you know that we are here to guide you, right? And definitely we will finish our syllabus in this game of NEET 2.0 as well. Okay. Done? So ready? Yes. Ready? Please tell me, ready? Okay, let's go. Let's start. Yes, let's go. Let's start. So the chapter name is what? The chapter name is what? It is Biomolecules. What is the chapter name, guys? The chapter name is Biomolecules. Okay, the word here is bio, means related to living organisms. So when you talk about the living organisms, definitely they are also made up of something, na? They are also made up of something. Here, before I'll relate this chapter with the evolution. Okay? When we discuss the evolution, when we talk about the evolution, we start our discussion with Big Bang theory. Yes or no? We start our discussion with Big Bang Theory. I hope you know about Big Bang Theory. Who gave this Big Bang Theory? Ebbe Lemaitre, right? Ebbe Lemaitre, he gave the Big Bang Theory, right? So we start our discussion with the Big Bang Theory, right? We talk about that explosion, that massive explosion, right, which we cannot measure in physical terms, and so we discuss about the origin of the universe, origin of Earth, origin of life. So many things are there. So many things are there. So when we talk about the evolution, I don't know whether you know it or not. You should know it. It is a part of your syllabus, of course. So we talk about the Operan and Haldane theory. Do you know that? We discuss about the Operan and Haldane theory, right? It is the most widely accepted when we talk about the origin of, right, when we talk about the origin of life and all, right? It is the most widely accepted theory, the naturalistic theory, Operan and Haldane theory. So here we talk about the chemogeny, right? Then we talk about the biogeny. Here, what do we discuss? We discuss about the chemogeny and we discuss about the biogeny. That is, firstly, there is a chemical evolution and then there is a biological evolution. So here we talk about the atoms, right? We discuss that how, these atoms all together form molecules, then bache elements will form, compounds will form, inorganic compounds, organic compounds, simple organic compounds, complex organic compounds will be there, and they all will aggregate, and they will, right, they all will aggregate and they are going to form the proto cells, protobionts. They are not exactly the living cells, but the structures from where the living cells arise. So basically what I'm trying to say here is that as per the conditions which were there on earlier Earth, reducing atmosphere type of conditions, high temperature type of conditions. So firstly, we talk about the chemical evolution and then we discuss about the biological evolution, right? We talk about the chemical evolution, then we talk about the biological evolution. And when we discuss the biological evolution, again, we talk about these organic compounds, right? So basically from this part, what I want to say, that even when you talk about a cell, right? Even when you people talk about a cell, right? The basic unit of life, the basic structural and functional unit of life. So here in the cell also, yes, bache, here in the cell also, this is what we discuss. We say the protoplasm is there, right? Your cytoplasm plus genetic material, that is making the protoplasm. Protoplasm is there, that's what we discuss. And here when you talk about the cell, it is also made up of these organic, inorganic things, right? So even, right, when you talk about the living organisms, you discuss about these elements, you discuss about these compounds. That's why we say that biology and chemistry, right, we have to go hand in hand. We have to go hand in hand. You know that if there will be no living reactions, if there will be no living reactions in our system, we are not going to survive, right? That is basically the metabolism. If there will be no chemical reactions, if there will be no breakdown and buildup of things in our body, we are literally not going to survive, right? If we are living means metabolism is there, chemical reaction is there. So chemicals, these compounds, they are important. So our body is made up of that. Let's take the example of a cell. Phospholipid bilayer. Proteins, after water you will see abundant. Proteins are abundant there, right? So basically, whatever these things are, we have to read it. We need to know about it. So it all comes under the biochemistry. And here in this chapter, this biomolecule chapter is also the part of biochemistry. That our body, our cells, they are made up of what? The reactions, right? Basically here in this chapter, you talk about the carbohydrates, the proteins, lipids, nucleic acid, enzymes, metabolism. That's what we are going to discuss here, right? That's what we are going to discuss here. So you should have a proper understanding of this chapter. It is one of the most important chapters, let me tell you, students. Okay, right? So that's what we have to discuss. Carbs, lipids, protein, nucleic acids, they are making our cell, they are the part of our cell. That's what we need to do. Okay, done? Yes, done? Okay, chaliye. So obviously, how can we forget NCERT? We'll keep our NCERT with us and we'll complete the chapter. What say? So guys, excited or not so excited? Dekho, if you are not excited, if you don't want to learn, if you don't want to know the things, then why to take this chapter? Why to discuss this chapter? I'll end the class, I'll put the recorded lecture. But if you are interested, I want you people to answer the questions. I want you people to interact here in the chat section. What say, guys? What say? You are not ready. You people are lying. You people are lying. You are like, nahi, yaar, now we don't want the marathons. Now we don't want to complete the syllabus, yaar. Nahi, ma'am. We are done. We are done. We'll again wake up in the month of February and then we will like how to crack the NEET examination in 1 month. We will watch that video and then we will start studying. Right now, still we have the time, but we are not going to utilize it. Nahi? We are not going to utilize it. Session duration, please. Rani, I cannot comment here. Matlab, it depends. It depends upon you. It depends upon me. Today, I cannot comment. After so long, yaar, we are meeting. Matlab, what do you think that? Matlab, what do you want? Ki yaar, ma'am, end the session in 2 hours or 3 hours. Nahi, yaar. Give some time. Give some time. Chaliye. So, there is a wide diversity in living organism in our biosphere, obviously, right? So, are all living organisms made up of same chemicals? Right? But there is a question here, ki all the living organisms, are we all made up of same kind of elements, same kind of compounds? So, they are saying that this is something that we have learned. The elemental analysis we have learned in chemistry. I know my students, they don't read anything seriously. So don't worry, we will discuss it here. So here they have given one, you can say that a kind of analysis is there, right? So basically when we start this chapter, we start with the chemical composition, that how to analyze the chemical composition. How do we start this chapter? We start the chapter with the chemical analysis, chemical, how to analyze the chemical composition. That's what you can say. So obviously we have to start the discussion as per that. And here you guys can see, if we perform such an analysis on a plant tissue, animal tissue or microbial paste, we obtain the list of elements like carbon, hydrogen, oxygen, nitrogen and several others with respect to their content per unit mass of a living tissue. So if same analysis is performed on a piece of earth crust, as an example of non-living matter, we obtain a similar list. So what you need to write, right? What you need to mention in your notes, number one, that when we do the elemental analysis, right, when we analyze the elements, elements in the living organism and in the living organisms, we are going to get same type of elements. Their composition can vary, but yeah, same kind of elements we are going to get in living matter or in non-living matter. So when it comes to the living matter, you can consider anything, right? You can consider the microbial paste, means you can consider the microorganisms, right, bache? You can even go for the plant material. You can even go for the animal material. So anything living, any paste you can see, right? You can say analyze their chemicals. You can analyze their elements. And when we compare it with the non-living matter, so here in your book, the example is given of earth crust. So when we compare it with the earth crust, we are almost going to get the same list. This is what they are saying. So all the right. So when there is a closer examination, it reveals the abundance of carbon and hydrogen with respect to other elements in the living organism and in the earth crust. So as I said, first of all, we are going to start with what? We are going to start with the analysis, right? Chemical analysis. So here you people can see, this is what you have to do. You have to make this flow chart, right? So firstly, we have to take the living tissue. What we need to do, bache? We need to take the living tissue. Again, you can take anything, microbial paste, plant material, animal material, whatever is available. You can take the plants, right? You can make their extract. You can take the animal. In the case of animal, just let's say a liver, any part of the body. This is what we have to take. This is what we have to take. Now we have to grind it. We have to grind it in this. What is it? It is your trichloroacetic acid. What's that? It is your trichloroacetic acid. It is a chemical. Which chemical it is? It is your trichloro. Yes, bache. It is your trichloroacetic acid. Right, bache? Right. So again, I'm making it in front, writing it in front of you. Yes, I am writing it in front of you. So we have to take the living tissue. We have to take the living tissue. You can take plant material. You can take animal material. Right, bache? And then living tissue which is grinded in acetic acid, which is grinded in trichloroacetic acid. See students, what is the formula of acetic acid? It is CH3COH. CH3COH. Yes. What's that? CH3COOH. Now it is trichloroacetic acid. So what are you going to replace? Remove this H and here it will be Cl, Cl and Cl. Right. So it is CCl3COOH. Yes. CCl3COOH. Or simply you can write so that is your trichloroacetic acid. So we have to grind the living tissue. We have to grind the living tissue in trichloroacetic acid using mortar and pestle. Using mortar and pestle. Okay, bache? And we have to make a slurry. What we have to do? We have to make a slurry. So firstly tell me, why are we using trichloroacetic acid here? Yes, why do we use trichloroacetic acid here? Why do we use trichloroacetic acid here? Yes, please tell me. Yes, please tell me. Yes, please. Why do we use trichloroacetic acid? Trichloroacetic acid. Bache, because this trichloroacetic acid, remember it's a weak acid and it is required for the precipitation. It can precipitate proteins, right? It can precipitate these compounds. It can precipitate proteins. That's why we are using the trichloroacetic acid, right? That is why we are using the trichloroacetic acid. So after it, what's going to happen? Yes, after this, what's going to happen? You know that, right? A slurry will be obtained. Yes or no? The thick slurry is obtained, right? We will get what? We will get the thick slurry, right? Now, after getting that thick slurry, what we have to do? A thick slurry is obtained. After that thick slurry, we have to strain it, right? It is strained through. It is strained through cheesecloth. Okay, you have to strain it. Just let's say you imagine in this particular beaker, you have this slurry, living tissue plus this trichloroacetic acid, right? Then we are going to strain it. So when you strain it, right, here you will get the filtrate and here you will get the retentate. What will you get? You will get the filtrate and the retentate. Am I right? What will you get? You will get the filtrate and you will get the retentate. So basically you will get two fractions here. Two fractions here. You are trying to filter out something. The something that will stay there in that cloth. That is the retentate. Whatever will come out, that is the filtrate. That's what I'm saying. Right? That's what I'm saying. So this filtrate, for this filtrate, remember one trick, that is fast. For this filtrate, remember one trick, that is fast. So what is the meaning here? That this filtrate is acid soluble. That this filtrate is acid soluble. And your retentate is acid insoluble. Your retentate is acid insoluble. Are you getting my point? So this one is acid soluble. This one is acid insoluble. One is acid soluble. One is acid insoluble. So that is what we have. Filtrate, retentate. Filtrate, retentate. Understood? Filtrate, retentate. Filtrate, retentate. So you will get two compositions. What will you get? You will get the two compositions here. Okay. Okay. So now when you talk about the filtrate, which is the acid soluble pool, right? Which is the acid soluble pool. Yes. So this filtrate, which is what? It is the acid soluble pool. It is soluble in acid and we are calling it as filtrate. The another fraction is of acid insoluble pool and that is your retentate. That is your retentate. Now, before this part, we'll discuss something. Dekho, basically we are talking about the biomolecules, right? These are making the living body. These biomolecules are present in our body. Yeah, they are making a living body. We are calling them as biomolecule. Like in NCERT, there is a line, all carbon containing compounds are biomolecule, right? But obviously that is not true. If you talk about a cell, I believe you know that, right? I believe you know that when you talk about a cell, in that cell, carbon containing compounds are there, organic compounds are there, but inorganic things are also there in our cytoplasm, right? They are also important there in our cytoplasm. So we cannot simply say that all carbon containing compounds are biomolecules. Yes, they are, but there are inorganic things as well. And now even when we talk about the biomolecules, right? Biomolecules that are the part of living body or that are making the living body. Here in biomolecules, you will further discuss two things. Like you will talk about biomicromolecules and there will be biomacromolecules. There will be biomicromolecules and biomacromolecules. Okay? Name the word is biomicromolecule. Micro, small, macro, large, of course. So biomicromolecules are the one whose molecular weight is less than 1000 Dalton. And here the molecular weight is above 10,000 Dalton. So basically, even in a living body, you will see some are monomers, some are that basic units. They are going to form the large units. They are going to form the biomacromolecules. They are going to form the polymers. That's what we have to discuss. So I'm explaining it here and then I will come back to that acid soluble part or insoluble part. But firstly, tell me, is it clear? So when you are talking about the biomicromolecules, their molecular weight is less than 1000 Dalton. Their molecular weight is less than 1000 Dalton. So here you can consider the example of monosaccharides, single sugars. We'll discuss it in today's class, like your glucose, like your fructose, right, etc. Right, bache. Then you can also talk about the amino acids. Right, your amino acids, they are biomicromolecules. Your nucleosides, your nucleotides, etc, they all are, because their molecular weight is less. They are not polymerized. They are biomicromolecule. When you talk about the biomacromolecule, their molecular weight is above 10,000 Dalton. So here you have polysaccharides. Poly means many, made up of many sugar subunits. Polysaccharides are there. Here you have proteins, you have nucleic acids, etc. So this part clear? Now, why are we talking about it? I'll tell you. See, we are discussing acid soluble pool and acid insoluble pool. We are discussing acid soluble pool and we are discussing acid insoluble pool. Right, bache? What are we talking here? So we are talking about the acid soluble pool and we are talking about the acid insoluble pool. Is that clear? So actually in your acid soluble pool, the substances, yes please write down, the substances with molecular weight less than 1000 Dalton. So yes, can I say that biomicromolecules are present in acid soluble pool? Yes, bache. So obviously, can I say that monomeric substances are found in filtrate? You tell me please. Can we say so? Monomeric substances are found in filtrate. Can we say so? Can we say so? Basically, we just completed the chemical analysis. I am talking about the acid soluble fraction. I am talking about the acid insoluble fraction. Before discussing that in detail, I told you when you talk about the biomolecules, they are the one that are making our living body. They are the one that are making our living body. Here in this chapter, polysaccharides, nucleic acid, proteins, lipids, enzymes, they will be discussed. Told you already. And I just told you that when you talk about the biomolecules, that's how you're going to divide biomicromolecules, biomacromolecules, biomicromolecules, biomacromolecules. So when it is biomicromolecules, the molecular weight is less than 1000 Dalton. When it is biomacromolecule, the molecular weight is above 10,000 Dalton, right? So basically the filtrate after this chemical analysis that we are going to get, it contains the acid, it contains the biomicromolecules, and not just the biomicromolecule. Please note down, it contains, yes, please write down, it contains inorganic compounds like sulfates, phosphates. It contains inorganic compounds, that is from NCERT, like sulfates and phosphates. Like sulfate and phosphates. Yes, like sulfate and phosphates, and thousands of organic compound. Right, bache? Right, bache? And thousands of organic compounds. So if you check, matlab from the in the NCERT it is given na ki from the chemistry point of view, if we check, so they have the different functional group. They are the nucleosides, nucleotides, right? They are the amino acid, etc., etc. Yes, etc., etc. So when it is the acid soluble pool na, it roughly represents what? It roughly represents the cytoplasmic composition. Right, that whatever is there in the cytoplasm, that will pass here in the filtrate. That's how it is. This part clear? This part clear? Yes? Now, when it is the acid soluble, obviously here you will get substances with molecular weight above 10,000 Dalton. Basically here you will see biomacromolecules. Jैसे you will see proteins, polysaccharides, nucleic acids, etc. But, right, nucleic acids. So one thing is, what I just said, that substances with molecular weight above 10,000 Dalton, except lipids. Except what? Except lipids. So lipids are biomicromolecules, right? We'll discuss this exception. Lipids are biomicromolecules, but still that lipids are the part of acid insoluble fraction. So substances with molecular weight above 10,000 Dalton, proteins, polysaccharide, nucleic acid, etc., except lipids. So lipids are having molecular weight in between 18 to 800 Daltons, right? In between 18 to 800 Daltons, right, bache? So substances with molecular weight above 10,000 Dalton, except lipids. Right, bache? Except lipids. Okay. Okay. So we can say that here we will see polymers. Right. What are you going to see in right hand side? Polymers. Again, the point is same, except lipids, because lipids are not strictly biomacromolecules. Please write down. Right, they are not strictly biomacromolecules. Please write down. Now the third point here is that it contains, just I told you, in the acid soluble pool, you will see the cytoplasmic composition, You will see the cytoplasmic composition, right, bache? And yes, this point I'm not going to explain because I have already explained here that here you are going to get protein, polysaccharides, nucleic acid and lipids. But lipids are not the biomacromolecules. So it represents macromolecules from, matlab if you talk about the composition of this retentate, it represents biomacromolecules from cytoplasm and organelles. That's the point. So it represents, filtrate is just showing us what? The cytoplasmic composition. The inorganic compounds also present in the cytoplasmic composition. But here you will see the biomacromolecules from cytoplasm as well as from the organelles. Right, bache? Right? Yes. Right? So now why do we say, and yes, what about these lipids? But again I'm repeating it, this is important. So when you talk about the lipids, right? When you talk about lipids, but lipids, their molecular weight is in range, their molecular weight is in the range 18 to 800 daltons, right? Their molecular weight is in the range 18 to 800 daltons. 18 to 800 daltons, right, bache? So obviously they are not biomacromolecules, but still in retentate. Why? Because lipids are, right? Why? Because lipids are insoluble in water. It is not soluble in water. So ma'am, what's going to happen? So when you are breaking a cell, these lipids, they are going to form some insoluble vesicles. Yes, they'll form some insoluble vesicles. So that vesicles will not pass there in the filtrate, they will stay in the retentate only. Just say, you know that cell membrane, phospholipid bilayer it is. So lipids are there, but they are insoluble in water. So when we break the cell component, these insoluble vesicles being insoluble in water, insoluble vesicles, they will not pass in the retentate, they will be there in the filtrate only. Is that clear? So this is about the lipids. Okay, is that clear? This is about the lipids. Okay, so they are still in retentate, but they are not biomacromolecule. So when you talk about the lipids, we are going to discuss it also, but here only I'm telling you, neutral lipids, they are having your alcohol plus fatty acids. They are making your lipids. Alcohol plus fatty acids, they are making your lipids. Is that clear? Okay, chale. So next is the analysis of inorganic compound. Okay, because not just the organic thing, but inorganic things are also there. So when it comes to the analysis of inorganic compound, bache, these are the steps here, right? Again, everything is given in NCERT. So what do you have to do? You have to take the living tissue again. Obviously, we have to burn it. Why? What's the requirement? You will burn it. What is going to happen? All the water will evaporate. Yes or no? All the water will evaporate, yes or no? Tell me please. I'm talking about the analysis of inorganic compounds. Yes, please. I am talking about the analysis of inorganic compound. Right. So obviously, firstly, you are going to take a living tissue. Yes. But yes, you have to check its weight as well. Right. So we have two weights. They say wet weight and dry weight. Obviously, even in ecology, we discussed that. Right. That wet weight, when the water composition is there, when the water content is there in that living tissue. Dry weight, when the water is not there. And when it comes to the biomass, right, we rely on the dry weight. Why? Because when it comes to the wet weight, you're talking about the water there. Water is present in that living tissue. That thing can vary, right? This thing can vary. Are you getting my point? This thing can vary. What can vary? This weight thing. Because as seasonal fluctuations can be there in that water. right? So we prefer the dry weight. So we will measure both dry weight, firstly wet weight, then we will evaporate the water and we will get the dry weight. Is that clear? Is that clear? So all the carbon compounds, right, because you are going to burn the tissue. So all the carbon compounds, what will happen? The water will be removed and all the carbon compounds will also be removed, yes or no? All the carbon compounds, you are burning the tissue completely, right? So water will evaporate. So when the tissue is fully burnt, the carbon compounds are oxidized to gaseous forms, isn't it? Isn't it? Carbon compounds are oxidized to gaseous form. Water vapors will also release, etc. So we are left with ash. We are left with ash here. So CO2, water vapor, they all will be removed and the remaining thing is the ash. And it is nothing but the inorganic elements, right? This ash is nothing but this ash is showing us what the content in the ash are: calcium, magnesium, sulfate, phosphate, etc. So in simple words, we can say that inorganic compounds are there in that ash. Right, inorganic compounds are there in that ash. So this is the analysis of inorganic compounds, right bache? This is the analysis of inorganic compounds. Yes, and last one is the overall chemical analysis. That's what you need to remember. So it is given in NCERT, and from this particular chapter, this particular chapter, the tables can come. Right, you need to decode the tables, right? You need to decipher that. You need to understand what information is there in that table. We will discuss it, okay? So here, see, chemical analysis is given. Yes, madam, we have completed it. So living tissue, grind it in trichloroacetic acid using a mortar and pestle, right? You will get a thick slurry. When we strain it through a cheesecloth or cotton cloth, you will get two fraction. One is filtrate, and filtrate is acid soluble. One is retentate, that is acid insoluble. Understood? So see this table. Look at this. Look at this. Please. Hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium and silicon. It is available. Now earth crust means non-living matter. Human body means living matter. So when you talk about it, in the earth crust and even in the human body, oxygen is abundant. Oxygen is abundant. That's what you need to understand. What is abundant in both? Oxygen is abundant. Yes, oxygen is abundant. Is that clear? Is that clear? Now, after oxygen, when you are talking about the earth crust, okay, after oxygen, what will you see? Silicon. But that silicon is negligible in human body. You should know about this information if you are targeting 360 out of 360 marks. So after this, ye dekho, look at this. Calcium is there, right? Calcium is there, right? After calcium, you will see sodium is there. After sodium, there will be magnesium is there. After magnesium, yes, what can we see? The sulfur and carbon, their composition is same, but hydrogen is there. Then this carbon and sulfur. Hydrogen is there. Then this carbon and sulfur. Okay, then this carbon and sulfur. But on the other hand, in human body, the silicon is negligible. In human body, silicon is negligible. You should know this. You should know this. And after oxygen, just say here, after oxygen, you will see silicon. After silicon, what will you see? You will see calcium is there. Calcium is there. Nitrogen is very little. Calcium is there. After that, the sodium, the magnesium, and then the hydrogen. But here, after oxygen in human body, in living body, what will you see? After oxygen, there will be carbon. After carbon, there will be, yes, bache. After carbon, there will be, look at the composition here. Nitrogen is there. Nitrogen is there, right? So firstly, oxygen, then carbon, then nitrogen, right? Then nitrogen, then calcium. So that is how you have to mark it. Got it? Got it? So you should know about these tables. Fine. You should know about these tables. Understood? Sure? Yes? Sure? Sure? Okay. So in higher classes, so this part is not required. So what you need to underline here that yes, then analytic techniques, they are saying ki whatever we apply to check the compound, to check the molecular formula, right? Obviously you will study that in higher classes. So this line, please mark it. All the carbon compounds that we get from living tissue can be called biomolecules. All the carbon compound that we get from the living tissue, we call from the carbon compounds we get from the living tissue can be called biomolecules. However, living organism has also got inorganic elements and compounds. So obviously it is not just the organic compounds that are there in biomolecules, inorganic compounds are also there. Right, bache? And that's why this experiment, that's why they're talking about this destructive experiment, ki check the small amount of living tissue, right? Then we have to measure its wet weight and then dry it, right? All the water will evaporate, remaining material will give the dry weight. Okay, when the tissue is fully burnt, all carbon compounds are oxidized to gaseous form. They'll be removed and the ash will be there. And thus ash, calcium, magnesium, all of it will be there. So inorganic compound like sulfate, phosphate, they are also seen in acid soluble fraction. So in the starting, when I mention acid soluble, insoluble, we have covered everything there. Okay, we have covered everything there. Okay, bache. So elemental. So earlier it was trichloroacetic acid wala thing. That is basically the chemical analysis. This is the elemental analysis. Right? This is the elemental analysis. Understood? This is the elemental analysis. Understood? Yes? Done? One minute. Okay, so elemental analysis gives elemental composition. Okay, so that's it. So now let's move to this part. Okay, ji? So obviously in the NCERT, the way this chapter is given, it is not basically good, right? Because they are randomly the things are there, right? Polysaccharides are there in the NCERT, but they are not talking about the monosaccharides. In your class 12th chemistry, it is given, but not in class 11. So we are going to complete everything here. We are not directly going to start from the polysaccharides. Rather, we will also talk about the monosaccharides. Okay? But before going forward, we are going to talk about something known as primary and secondary metabolites. Okay, that's what we need to discuss, that what is the meaning of primary and secondary metabolites. See students, we all know one thing, right? We all know one thing, that being living means, this line is even here in this chapter, that being living means certain conditions need to be fulfilled. We call it as the defining property, right? And metabolism is one of them. Metabolism is one of them. When we talk about the metabolism, it is having two things. It is having catabolism and anabolism. It is having catabolism and anabolism. So cut, cut. Right? To cut, to break. That's how you have to remember. So catabolism means break down. Anabolism means build up. Catabolism means break down. Anabolism means build up. Right? So firstly, the word is metabolism. Right? The chemical reactions which are going on in our body, and they are keeping us alive. They are keeping us alive, right? Because in our body there is these reactions are there where something will make, something will break. For an example, we are eating the food. Why? Because we will get energy from that food. Let's say we are getting glucose, so we have to oxidize it, right? So oxidation of glucose in multiple steps, right, will release the energy, that energy will be trapped in the form of ATP, and this is the energy currency of our cell. That's how our system works. Right, bache? Right, bache? So obviously, if this glucose will not get oxidized, how are we going to get our energy? Right? How are we going to get our energy? Are you getting my point? So chemical reactions are important. Let's take this. So it is a kind of catabolic reaction, right, where the breakdown of glucose is there. But when you discuss it in respiration, we say that respiration is amphibolic. Right, because multiple steps are there. Sometimes there is build-up, sometimes there is breakup, but that's a different story. So the photosynthesis, anabolic, the build-up is there. Carbon dioxide, water, they are giving us glucose. Right, they are giving us glucose. Are you getting my point? So metabolism is something very important in our body. So basically we have a lot of biomolecules. One will get converted into another, that another one will get converted into another one. It is as per our body's need. Okay? So when these multiple steps are there, right, the chemical reactions are going on. In between, some intermediates are also there. So those which are participating in this, we are also calling them as metabolites. Right, these compounds, these are the metabolites which are there, which are present there. Right, these are what? These are metabolites. Is that clear? Is that clear? So now we are talking about the metabolites. We are talking about the primary and secondary metabolites. What is this? Means these are the compounds. Basically, in simple words, if I have to say, obviously these are the compounds which are present in our living body. Yes, these are the compounds which are present in our living body. Yes or no? These are the compounds that are present in our living body. Yes or no? Yes or no? Obviously they are, right? Something is primary, something is secondary. Obviously primary, priorities, matlab it is more important, or you can say it is directly playing role in our system. That's how you can remember. That's how you can remember. We are talking about the metabolites. Right, obviously these are the chemical compounds which are there in our body, right? During metabolism they are forming, they are getting converted into another form. That's how you can keep it in your mind. So these compounds, these chemical compounds, we are saying some are primary, some are secondary. Some are primary, some are secondary. Right, what are we saying? Some are primary, some are secondary. That's what we are saying. Primary, priorities, means they are the most important one, means they are, right, means they are somewhere playing direct role in our system or in our system's functioning. That's how I can remember. Secondary, they are important, but maybe their role is not that clear. That's how you can remember. But actually they are like that only. I'll explain in very simple words. जैसे ये देखो, the most exciting aspect of chemistry deals with isolating thousands of compounds, small, big, from living organism, and to check their structure. ठीक है ma'am? Okay. Now what are they saying? That from living organisms, thousands of organic compounds are obtained. Amino acids, sugars, etc., etc. Right, bache? So all these biomolecules are what? They are metabolites. All these biomolecules are what, bache? Yes. All these biomolecules are what? They are metabolites. Yes or no? Tell me. Yes or no? Tell me. Yes or no? Tell me. Thank you, Shivam. Thank you so much, Shivam Bache. Chalo, now focus here. So all these biomolecules are what? They are metabolites. So in animal tissues, one notices the presence of all such categories of compound, right? So these are the primary metabolites. So when you are saying primary, they are having the direct role, right? They are having the direct role. Wait, let me mark this. Let me mark this here in NCERT only. When you are talking about the primary metabolites, please mark it. Kindly mark it. While primary metabolites have identifiable functions, they play known roles in normal physiological processes, right? What are primary metabolites? They have identifiable function, means we can identify their function. We can say, okay, this is how they are involved in our bodies, right? Physiological processes. But when you talk about the, yes, when you talk about the secondary metabolites, right? They are not having that identifiable role, but they are important for the host organism. Like from plants, from fungus, we get so many things. From plants, we get alkaloids, right? We get flavonoids, we get different kinds of, toxins, essential oils, etc., etc. They all are what? They all are secondary metabolite. They all are what? They all are secondary metabolite, right? So primary metabolites are the one that are having the identifiable function, and here you people can see, right bache, that one analyzed plants, fungus and microbial cell will see thousands of compound other than these primary metabolites. So primary metabolites mein, jaise you have, yes, what do you have in your primary metabolites? Please tell me. What do you have in your primary metabolites? You have amino acids, sugar, chlorophylls, etc., etc. Right? Right? So they are playing role in photosynthesis, respiration, in protein and lipid metabolism. So primary metabolites may you have these amino acids, you have sugars, you have the various kind of pigment. You have amino acids, you have sugar, you have the chlorophyll, you have the hemes. All of them are making the primary metabolite. Is that clear? So sugar, amino acids, lipids, nucleotides, nucleosides. Right, you can write it here. So nucleotides, nucleosides, hemes, all of it, etc. They are having a direct role. These are the primary metabolites. But these alkaloids, flavonoids, rubber, essential oils, antibiotics, that colored pigments, scents, gums, they all are secondary metabolites. Okay, so some secondary metabolites, you know that many of them are useful to the human welfare, right? And some have ecological importance. So from the chapter biomolecule, it is one of the most important tables, right? It is important. Understood? It is important. Understood? Understood? It is important. Yes, Vishnu. Okay, so examples are important. Pigments, carotenoids, anthocyanins. Anthocyanin is water soluble. Carotenoids, fat soluble. You should know them. You should know the examples. Then alkaloids, morphine, codeine. Right, morphine you know na? It is analgesic. Analgesic means pain reliever. Analgesic means pain reliever. Codeine, right? They are having kind of same function. Codeine is there in our cough syrups. You know that? In the cough syrup, the codeine is present. Do you know about it? Yes. Do you know about it? You should know about their roles. You should understand it. Yes, please. Done? Right? So obviously they are what? They are the analgesics. Analgesics means pain reliever. Your morphine. Morphine. Yes, obtained from poppy plant, and the codeine. They both are analgesic. Right? They both are analgesic. Is that clear? Is that clear? So they are, the codeine is a, yes. So it is also used to treat the pain. Okay? It is also used to treat the pain, to manage the pain. Okay? And to treat the cough as well. Fine? Right? So with combination with various things, you know, they are used in the medicine. So you should know about it. Then terpenoids, monoterpenes, diterpenes, right? That is also a secondary metabolite. That is what? That is also secondary metabolite. Is that clear? Terpenes, right? Yes, bache. Terpenes, they are having isoperine. Okay, so they are used for flavors, right? For in perfumes and all, they are used, right? So they are also having the different roles. Okay, they have that aroma. Fine, they have that aroma. Okay, yes, yes. So they have different types of monoterpenes, diterpenes. So you know camphor. Do you know camphor? Yes. Do you know camphor? Camphor, camphor, camphor. Camphor is also an example of terpene. Camphor is also an example of terpene. Done? So they also sometimes help in, you know, reducing the pain, to fight stress, anxiety. So many examples are there. Okay? Many examples are there when you talk about the terpenes. Right, bache? Yes. So they are even commercially derived. Fine. Fine. Sure. Okay. Then essential oils में lemongrass oil, then toxins में heparin, ricin, right? Lectins, concanavalin A. Examples are important, right? बच्चे, examples are important. So tell me, what are the lectins, right? What are the lectins? Tell me quickly. What are the lectins? What are the lectins? Again, bache, they are the proteins. These proteins are attached to carbohydrates. They play different roles. Jaise cell to cell recognition mein they are playing the role, and during cell signaling, right? During cell signaling, during conveying that, right? These lectins are playing the role, bache, right? Bache, so cell to cell interaction mein, in that cell signaling, they have different roles, right? Bache, so these are the proteins that are attached to what? That are attached to carbohydrates. Okay. Okay. Done. So your beans, bell peppers, they all are having lectins, right? They play different roles. Okay. Sometimes even fight with the infection. So drugs may vinblastine. So then this vinblastine is obtained from your vinca rosea. It is obtained from your vinca rosea. Very common type of plant. Do check it. Vinca rosea. Catharanthus. Right. Apocynaceae is the family to which it belongs. I still need to check, but I think it is the Apocynaceae family. So vinca rosea, do check it. Right, so this vinblastine is obtained from it. That is also anti-cancerous. Right, this vinblastine is what? It is anti-cancerous. Then bache, curcumin, etc. They all are the drugs. Right, so polymeric substances mein rubber, gum, cellulose, all of it is obtained. So any doubt here? Yes, any doubt? Please tell me. This table is very important. This table is very important. Please tell me. Yes. So these are the drugs. So vinblastine is anti-cancerous, and curcumin, do you know it is obtained from turmeric? It is obtained from turmeric. This curcumin, turmeric, it is having anti-inflammatory properties. So you know na, turmeric, right? It is medicinal. So that's how it is. So you should know about it, right? You should know about it. So in the paper, they can also give match the column type of questions. They can also ask you what is a lectin, examples of lectin, role of lectin. That's how they can ask. So this is important. Okay, this is important. Done? Sure. Sure. Good. So now, what are we going to start? We are going to start carbohydrates. What are we going to start, students? Now we are going to start carbohydrates. Okay? Carbohydrates. What are we going to start? We are going to start carbohydrates. Okay? Yes. Right? In my chat section, the proposal thing is going on. Vishnu Singh, because I won't marry with you. First of all, bache, work on your grammar. Okay? And why such kind of conversation is going on? Yeah, don't waste your time. Don't waste your time. Do something productive. Leave it. Leave it. Seriously, leave it. Why do waste time in all that things, yeah? Trust me, you have time. You have time for all that things. Leave it. Literally, leave it. Marriage is a very big responsibility. Avoid it as much as you can. So carbohydrates, hydrates of carbon. Carbohydrates, hydrates of carbon. Now what are carbohydrates? What is the meaning here? Tell me. What are carbohydrates? What is the meaning here? Obviously, when we hear the word carbohydrates, obviously we are getting the energy from these carbs, right? It is our main respiratory substrate. Like we are talking about the glucose. Glucose is an example of what? Glucose is an example of carbohydrate, of course. Glucose is what? Glucose is an example of carbohydrates, right? The respiratory substrate, it will be hydrolyzed, it will be oxidized, it will give us energy. It will be oxidized, it will give us energy. Carbohydrates, right? The fruits, they are having the carbohydrates, right? In our meal, basically, we want the food, the energy. So that's how we are going to digest it. This is what we know when we hear the word. This is what we know when we hear the word carbohydrates, isn't it? Isn't it? So one thing is here, that what are these carbohydrates? If I have to define it, obviously they are also the organic compounds, right? What are they? They are obviously, they are also the organic compounds having carbon, having hydrogen, oxygen, and not just the carbon, hydrogen and oxygen, and some contain nitrogen and sulfur. Yes. So they are the organic compounds. They are the organic compounds having what? Having carbon, hydrogen and oxygen, and some even contain nitrogen and sulfur. Some even contain nitrogen and sulfur. Now, why do we call them as the hydrates of carbon? See, when you talk about the carbohydrates, CHO, that's what we have, CHO. So one part of carbon, two parts of water, one part of oxygen is there. So basically, when you look at this HO, just like water, water is H2O, two parts of hydrogen, one part of oxygen. So even in the carbohydrates, this is what you people are going to see. Right, this is what you people are going to see. Two parts hydrogen, one part oxygen, just like water. Two parts hydrogen, one part oxygen, just like water. Is that clear? Is that clear? So in simple words, you can write it that in carbohydrates, the ratio of carbon, hydrogen and oxygen is 1:2:1. So hydrogen and oxygen is in 2:1 ratio, right bache? Is in 2:1 ratio. Yes or no? Yes or no? So they are the hydrates of carbon, right? They are the hydrates of carbon. So chemically, what are they? Chemically, what are they? If chemically I have to define carbohydrates, then you know these chemically they are polyhydroxyaldehyde or polyhydroxy ketones. What are they? They are polyhydroxyaldehyde, or what are they? They are polyhydroxyaldehyde or polyhydroxy ketones, or compounds which on hydrolysis yield such kind of compounds. Okay? Or compounds which on hydrolysis are going to provide us this. Okay? So carbohydrates, chemically polyhydroxyaldehyde or ketone. Is that clear? Is that clear? Yes, is that clear? Yes, please. Yes, please. Polyhydroxyaldehyde or polyhydroxyketone, or the compounds which on hydrolysis are going to yield polyhydroxyaldehyde or ketone. That's how you can define. So what is the word? Poly. Poly means many. Many. When the word is hydroxy here, remember you are talking about the OH group, right? So polyhydroxyaldehyde or polyhydroxyketone. So when I'm talking about this, right, means in the case of carbohydrate, the functional group can be aldehyde or it can be ketone. It can be aldehyde or it can be ketone. And let me tell you, students, see here in this particular chapter, no doubt the monosaccharide, oligosaccharides are not explained in the chapter, but they are important to understand what polysaccharides is. They are important to understand what glycosidic bond is. So it's a request. Please do not consider it as out of NCERT. It is in NCERT. It is the part of your syllabus. It is somehow going to help you in the chemistry. Okay, so please pay attention. So we are saying organic compounds having carbon, hydrogen, oxygen, and they can also contain nitrogen and sulfur. Plus I'm saying that here the ratio of carbon, hydrogen and oxygen is 1:2:1, right? So chemically they are what? Chemically they are polyhydroxyaldehyde, they are polyhydroxy ketone. Chemically they are polyhydroxyaldehyde, they are polyhydroxy ketone, and they have, so poly means many, hydroxy means OH, that's what they have. Right, poly means many, hydroxy means OH. That's what we are going to see here. So they have two functional groups, aldehyde as well as the ketone, aldehyde as well as the ketone, aldehyde as well as the ketone, and we know that it is our principal source of energy. It is our principal source of energy, right? So we will discuss it in the form of aldose, ketones as well, but now focus here. So how do we divide these carbohydrates? How do we divide it? Please write it. So what are you talking? You are talking about the carbohydrates. What are you talking? You are talking about the carbohydrates. So on the basis of hydrolysis product, that's how we are going to divide it. One second. On the basis of hydrolysis product, we are going to divide it. जैसे we have monosaccharides, we have oligosaccharides, and we have polysaccharides. So we have mono, we have oligo, and we have polysaccharides. We have monosaccharides, we have oligosaccharides, and we have polysaccharides. So it is the carbohydrate, main source of energy, and our energy basically we are getting from them. Okay? Yes. So what is monosaccharides? They cannot be hydrolyzed further into smaller carbohydrates. They are the simple type of carbohydrates. Mono means single, saccharides means sugar. Mono means single, saccharides means sugar. So these are the simplest type of carbohydrates that cannot be, yes, hydrolyzed further, that cannot be hydrolyzed further into smaller carbohydrates. Into smaller carbohydrates. Done. Monosaccharides. Monosaccharides, we will discuss that in detail. Yes. So oligosaccharides, these are formed by the condensation of, these are formed by the condensation of, yes, these are formed by the condensation of 2 to 10 monosaccharides unit. So basically 2 to 10 monosaccharides. So when I'm saying monosaccharides, monosaccharides माने single sugar. When I'm saying monosaccharides माने single. Saccharides means sugar. Monomers they are. Like your glucose, like your fructose, right? Like your glucose, like your fructose, like your galactose, right? Like your mannose. They are what? They are the monosaccharides. They are the monosaccharides. So tell me, where do you see monosaccharides? In the acid soluble pool or in the acid insoluble pool? Where will you find it? In the acid soluble pool or in the acid insoluble pool? Please tell me. In the acid soluble pool or in the acid insoluble pool? In the acid soluble pool, in the acid insoluble pool. Yes, please. I'm talking about the monosaccharides. I'm talking about the monosaccharides. Okay, so monosaccharides will be there in the, because it is a It is a monomer. It will be there in the acid soluble pool. So when you talk about the polysaccharides, they will be in the acid insoluble pool, and they are formed by the condensation of more than 10 monosaccharides. More than 10 monosaccharides subunits will join, they'll result in the polysaccharides. More than 10, there'll be many, they will be many, right? There'll be many. So this is about the carbohydrates on the basis of hydrolysis product. So the very first thing that we need to start is monosaccharides. Okay, we will talk about what? We will talk about the monosaccharides. So what I just said, that they are not, they will not further hydrolyzed, right? They are not further hydrolyzed. That is what I'm saying. They are not further hydrolyzed. That is what I'm saying. So what is the word? The word is monosaccharide. So as I said, they are the simplest sugar. They are the simplest sugar, right, bache? So when you talk about their general formula, it is CnH2nOn. CnH2nOn, right? It is their general formula, CnH2nOn. I'll give you a very simple example, glucose. Okay? I'll give you a very simple example of glucose. In the glucose, N is the number of carbon, right? So N here is 6 in the case of glucose. So C6, H will be C6H12O6, C6H12O6. So it is the general formula. It is the general formula. Yes, bache. Yes, bache. So N is the number of carbon. Here you can even write Y, and you can say that Y is the number of oxygen there. Okay, ji? So we are talking about what? We are talking about the monosaccharides, and I am saying they are not further hydrolyzed into smaller carbohydrates. The empirical formula, your general formula is CnH2nOn. Or how can you write it? CnH2n, because carbohydrates are hydrates of water, and CnH2ON, that's how you can write it. That's how you can write it, right, bache? Right? So it is not further hydrolyzed into the sugars, But bache, remember, right? That here these monosaccharides, when it comes to the carbon atoms, that how many carbon atoms can be there in a monosaccharide? Carbon atom is 2 to 7. Carbon atom is 2 to 7. Just say, it's like, if I have to give you an example, just say, let's say this brick. Just an example, let's say this brick is of 5 centimeter. You have another brick that is of 10 centimeter, yeah, 15 centimeter. Let's take the example. You have one brick only. One is 5 centimeter, another is 10 centimeter. So basically, when you are talking about the monosaccharide, it will be the single sugar only. It is the single sugar only, but the number of carbon can vary, right? It can be, it can have 3 carbons, 4, 5, 6 or 7. So 3 to 7 carbons, right? What will you see here? You will see 3 to 7 carbons, right? What are you going to see here? 3 to 7 carbon. Is that clear? Is that clear? Oh, by mistake I'm mentioning, I wrote 2 here. It is 3 to 7. Understood? It is 3 to 7. Understood? Understood? Yes? Yes? So if I talk about the properties of these monosaccharides, so you can even write they are highly soluble in water, right? They are the one that are highly soluble in water, plus they are sweet in taste, right? Not all carbohydrates are sweet in taste. Your polysaccharides are not sweet in taste. They are not soluble in water. But the monosaccharides, they are so highly soluble in water. They are sweet in taste, and they are having reducing nature. We will talk about the reducing nature thing, right? Various tests are conducted on the basis of that. So they have reducing nature, right? They have reducing nature, right, bache? Right? So they can reduce your sorry for that. Okay, so they can reduce the Cu2 plus into Cu plus ions. Okay, they can reduce the Cu2 positive into Cu plus ion. Okay. Okay. Yes. Right. right? So Cu2 positive into Cu positive. So basically cupric into cuprous, am I right? Cupric into cuprous, है ना? Cupric into cuprous. Yes, Cu2 positive is cupric to cuprous, and that is the basis of many tests, right? Jैसे I'll discuss its reducing nature. It is important. I'll discuss it. Okay, so this part clear. This part clear. So monosaccharides can occur in two forms, right? Monosaccharides can occur in two forms. In how many forms, बच्चे? It can occur in two forms. जैसे straight chain and ring form. Straight chain, second is ring form. So when you talk about this straight chain, it is also known as Fischer projection. Fischer projection. Again, I'll give you the example of glucose. So I'll suggest you to remember the structure of glucose, galactose, fructose, ribose, deoxyribose. That will be sufficient. Glucose, galactose, fructose, ribose, deoxyribose. So do remember the structure. Do remember the structure, right? Yes, do remember the structure. So straight chain, your Fischer projection. Again, as I said, I'll give you the example of glucose. See, so glucose is having 6 carbons now. 1, 2, 3, 4, 5, 6. Right? 1, 2, 3, 4, 5, 6. Okay? So here you have, chalo, let's say C double bond OH. So 1, 2, 3, 4. So H, OH, OH, H, H, OH, H, OH, H, H and OH. So see this, right? Your glucose. So we just said now that chemically carbohydrates are poly means many. Polyhydroxyaldehyde, polyhydroxyketone. Polyhydroxyaldehyde, polyhydroxyketone. So they have many hydroxyl group, many OH groups are present. So straight chain. So as I said, they are also having the ring form. So when you talk about the ring form, you have two things. One is pyranose form and another is furanose form. Furanose form, 4, right? So basically in this ring, 4 carbon atoms, 1 oxygen, and in the pyranose, 5 carbon atoms and 1 oxygen. So your monosaccharides can occur in these two forms. So straight chain, Fischer projection, ring form में pyranose and furanose is there. Pyranose and furanose is there. Okay, what is there? Pyranose and furanose is there. Now glucose. Again, let's take the example of glucose. Let's take the example of glucose. Again, bache, honestly speaking, this is very interesting to discuss about this ring formation, that how from straight chain the ring forms, right? What about that bonds and everything? I personally like it a lot, right? Biochemistry is my favorite. Biochemistry is literally, the subject is my favorite, but obviously time crunch is also there. Plus you are preparing for an exam, so we have to focus on that exam only, right? So that's why I am not able to discuss it, but whatever can simplify your topic, we are going to discuss that. ठीक है जी? So HOH, HOH, OHH, HOH. So CH2OH. So what's that? It is your glucose. What's that? It is your glucose, the ring form of glucose. So this ring form is what? It is pyranose. Say, see, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So 5 carbon in a ring, 1 oxygen is also there, right? So carbon 1, 2, 3, 4, 5, and this is the 6th carbon, but I am talking about the ring here, right? This is pyranose. 5 carbon in the ring, 1 oxygen is there, right? Your glucose, right? And the another thing is, see, what's that? Anybody here in the class? Yes, this is your ribose. Yes, even you need to remember this, ribose. So look at this. It's a furanose form. It's furanose, right? So you can see this carbon number 1, carbon number 2, carbon number 3, carbon number 4. So in the ring, 4 carbon, 1 oxygen, and ribose is a pentose. So here you have the 5th carbon. Is that clear? Is that clear? So 2 ring forms clear, straight chain clear. So that's how your monosaccharides are present, right? That's how your monosaccharides are present. So is it clear? Yes. So the structure of glucose, galactose, both in the ring form and even in the straight chain form, it should be on your fingertips, right, bache? So when you talk about the monosaccharides, I told you that the number of carbon, right? So when you talk about the monosaccharides, I told you that when it comes to the number of carbon is 3 to 7. So if it is 3 to 7, 3 carbons, triose will be the word, right? So I'm writing it like this only, NS3. NS3 matlab number of carbon. 4 carbon, tetrose. 5 carbon, pentose. 6 carbon, hexose, right? 7 carbon, heptose. So obviously this is your monosaccharide, but when you hydrolyze it further, you are not going to, right? When you hydrolyze it further, you are not going to get what? You are not going to see any further monosaccharide. ठीक है जी? So tri, tetra, penta, hexo and hepta, right? Triose, tetrose, pentose, hexose and heptose. Like your glucose is having 6 carbon, glucose is hexose. Right? And now when we define it chemically, I just told you, right? Polyhydroxyaldehyde, polyhydroxyketone. So aldehyde and ketones, what are they? Aldehydes and ketones, what are they? Obviously it is the functional group. Aldehyde and ketones, what are they? Functional group. So aldehyde is your C double bond O. C double bond OH. That's your aldehyde. Ketone is also C double bond O, like this, right? C double bond O. So when you look at both the functional group, they are having the C double bond O, they are having the C double bond. So any functional group can be there. It can be aldehyde, it can be ketone. It can be aldehyde, it can be ketone. So if it is the aldehyde, C double bond O. If it is the ketone, C double bond O. So basically in both the functional groups, you have this carbonyl group, C double bond O, carbonyl group. That's a previous year question. And I believe you know this, right? That your aldehyde is a terminal group. It will join at the end of the chain. And this ketone is subterminal. So it will not be there at the end. That is subterminal. I'll give you the example here also, right? So if let's say your functional group present is in the carbohydrate is aldehyde, you will use the word aldose. If the functional group is the ketone, you will use the word ketose. Wait. जैसे ये देखो, the examples are given. Triose, tetrose, pentose, hexose, heptose. So glyceraldehyde, erythrose, these are the examples given. So now I'll tell you what is aldose and what is ketose. Then let's discuss it. Aldose or ketose. So aldose when the functional group is aldehyde. Ketone when the functional group is ketone. So जैसे let's say if n is equals to 3, if n is equals to 4, if it is equals to 5, 6 and 7. You have the examples and you can relate. I'll start with the simplest one. 6 carbon glucose, and you know that you have the general formula CnH2nOn or CnH2ON, right? You can use this general formula and you can check it. So glucose, right, is an aldose. But when you talk about the fructose is the isomer of glucose now. So glucose is also having 6 carbon, fructose is also having 6 carbon, glucose is also having 6 carbons, fructose is also having 6 carbon. But fructose is a ketose, glucose is an aldose. Fructose is a ketose, glucose is an aldose. Okay? Okay? Yes? Right? Okay, chalo, one more example. Let's start with the this glyceraldehyde, that's a monosaccharide. Your dihydroxyacetone, that's a ketone, the functional group here. Okay, then you have N is equals to 4. Erythrose. Just say there is one example, erythrose. Erythrose. Now, focus on the nomenclature. Erythrose is aldose, means functional group is aldehyde. If it is erythryl, ulose. Erythulose. Just say I'll give you one more example. Right, I'll give you one more example. Ribose, you know that it is in RNA, and we also know about this ribulose. Remember rubisco? Do you remember rubisco, ribulose? Yes, we have completed the plant physiology now. So ribulose, bisphosphate. Yes, ribulose, bisphosphate, carboxylase, oxygenase. Ribulose, bisphosphate, carboxylase, oxygenase, rubisco, ribulose. So if it is simple ribose, you are talking about the aldose. Functional group is aldehyde. If it is ribulose, it is functional group is ketose. Functional group is ketose. Matlab, functional group is ketone, so it is a ketose. Right? It is a ketose. Understood? Right? So here you can write down ribose, yeah, xylose, ribulose, right? So glucose, fructose, yeah, yes, even the mannose, galactose is also there. And this is pseudohaptose, and this one is pseudoheptulose. Ul word will be added, right? What will be added, bache? Ul will be added. What will be added? Ul will be added. What will be added? Ul will be added. Understood? Understood? Sure? Sure? Okay? Okay. So this is about the functional group. Acha, one more thing. Now let's say, if in the paper they are simply giving you ribose, what is the formula of ribose? Obviously, N is equals to 5. If you know that, put it in formula. Put it in formula. So you will be like HC5, H10, O5. Right? And that's even a PYQ. That is also a PYQ. So remember one thing. Just say in the DNA. Firstly, let's see this, then I'll discuss. Here, look at this. An aldotriose, glyceraldehyde. Aldose it is. So it is attached to the ends, HOH this. Right? 1, 2, 3, 4. It is also aldose. Right? The functional group is terminal. Aldehyde it is. So when it is subterminal, ketose, C double bond O. Right? C double bond O. Yes. So it is C double bond O. Hexose it is. Right? Carbon number is 6. Carbon number is 6 here. Carbon number is 6 here. Yes, carbon number is 6 here. Okay. So see this. This is the structure of fructose. CH2OH, right? Third carbon OH is on this side. HOH, HOH, this. So glucose and fructose. Look at the glucose, look at the fructose, and that's it. That's all you should know about their structure. So aldehyde group, ketone group. Aldehyde terminal, ketone is subterminal. Okay? And see this. Now, okay. So do you know about alpha glucose and the beta glucose? Do you know anything about alpha glucose and the beta glucose? Alpha glucose and the beta glucose? It's very simple. Carbon number 1. Carbon number 1. Carbon number 1. Look at the structure of glucose. Look at the structure of glucose. Let me draw this. It's a pyranose. HOH, first carbon, second carbon. When it is the third carbon, OH will be on this side, this side. Okay. That's how it is. Carbon number 1, carbon number 2, carbon number 3, 4, 5 and 6. Third carbon. Look at the position of OH. Now this is your ring. Carbon number 1, carbon number 2, carbon number 3. Look at the position of OH here. This is 4th, this is 5th. Okay, and this is obviously the 6th carbon. Now look at the first carbon. That's how you are going to remember it. As I said, obviously we are not going to discuss it in too detail, but whatever is required for the understanding of the chapter, we have to discuss that. Okay? So first carbon HOH, first carbon HOH, first carbon HOH is there. Now the point here is carbon number is 1. So if hydrogen above, that's how I remember. If hydrogen above, you have to see this first carbon only. It will be alpha glucose. It will be alpha glucose. It will be alpha glucose. Now, at the same carbon, if it is like this, so if it is like this, the first carbon, if it is like this, the first carbon, look at this, hydrogen below, beta glucose, then it will be beta glucose. Alpha beta clear? Alpha beta clear? Yes, alpha beta clear? Alpha beta clear? Sure? Are you sure? ये देखो, right? Alpha D glucose. Okay, that's your homework. If your chemistry is good, you can tell why this D is there. This is sometimes plus sign, sometimes minus sign, sometimes D, sometimes L. The way it is written, sometimes they are written like this, sometimes they are written like this. What's the difference in that? Right? What's the difference in that? That's what you have to tell me. That's what you have to mention in the chat section, in the comment section after the class. Okay, in the comment section after the class. So alpha D glucose, beta D glucose. Right? Alpha D glucose, beta D glucose. Okay? 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 Next, you know that ribose, right? You should know about the structure. Ribose it is, right? It's a pentose, 5 carbon containing sugar. And we know that it's the important part of nucleic acid. Which nucleic acid? Your RNA. Your RNA is having this. In this chapter itself, we will discuss it. We'll discuss it here only. Okay? Then you have deoxyribose. Deoxyribose. And this deoxyribose is in the DNA. Right? Now, when you look at the ribose and deoxyribose, first of all, tell me, is it alpha ribose or beta? Is it alpha or is it beta? Is it alpha or is it beta? Is it alpha or is it beta? Obviously both are beta. Why? Hydrogen is below, OH is above. So they are beta. This one is also beta. So look at the structure of ribose. Look at the structure of deoxyribose. So when you talk about the deoxyribose, deoxy without oxygen. So at the second carbon, at second 2' carbon, your second carbon, O is missing. Oxygen is not there. Right? You have to be specific. When you compare ribose with deoxyribose, you people will see that at second carbon, right? Here in ribose, OH is there, but here it will only be H. And obviously, on the evolutionary point of view, the significance is there. It is related to the reactivity or the stable nature of the DNA that you will understand in the molecular basis of inheritance. Okay, so it is the deoxyribose. So obviously students, if it is ribose, I told you already the formula C5H10O5, and here in deoxyribose only one oxygen is less, so it will be C5H10O4. So bache, this is an exception, right? So deoxyribose, it doesn't follow the general formula. It's a monosaccharide that doesn't follow the general formula. It doesn't follow the general formula. Understood? It doesn't follow the general formula. Understood? Pakka? Sure? Sure? Right? So monosaccharides clear. Monosaccharides clear. Jैसे we have glucose, galactose and all. अरे हाँ. Okay. Done? Any doubt? Any doubt? Any doubt? Right? So when you say alpha and beta forms, you are talking about the anomers, right? You are talking about the anomers. जैसे we say ना कि glucose occurs in cyclic structure, position of H and OH groups are changed on first carbon, so they are the anomers. So alpha, beta, glucose, they are the anomers. Now I will tell you what epimers are. Now I will tell you what epimers are. Okay? So do you know anything about epimers? Do you know anything about epimers? So epimers are also the kind of isomers. Right? Isomers, you understand? Isomer is same one माने molecular structure, same molecular structure but structure में, same molecular formula but structure में vary. है ना? Same molecular formula but structure में vary. Yes or no? Same molecular formula yes? So they are the isomers, they are formed, right? They are the isomers. Yes, they are very interesting epimers. They formed as a result of interchange of H and OH group around carbon 2, 3, 4. Let me explain this. It's very simple. I hope you all know about the glucose, right? I hope you all know about the structure of glucose. Hanji, you know about it or not? You know about it or not? Please tell me, do you know about it? If not, then please tell me. Yes. If not, please tell me. Right? And if you want to write it, you can also write it in this way. Ki C double bond OH is there. Matlab, it's all up to you. Because it is ultimately showing the aldehyde group. That's what we have. See, this is your glucose, yes or no? This is your glucose, yes or no? Carbon 1, 2, 3, 4, 5, 6. This is your glucose, yes or no? है ना? Yes, ma'am. ये देखो. Now it is mannose. It is mannose. What's that? It is mannose, right? Carbon 1, carbon 2, carbon. See, at carbon number 2, मतलब rest everything is same. Rest everything is same, right? When you compare it with the glucose, rest everything is same. But at second carbon, it is mannose, then the carbon number will be second. At second carbon, look at the position of H and OH. It is interchanged, right? It is interchanged. Look at the position of H and OH. Is it interchanged? है ना? And now look at this part. This is your galactose. This is your galactose. So this is your galactose. Look at the fourth carbon here. Look at the fourth carbon here. So rest everything is same, but at fourth carbon, OH and H is interchanged. So they are epimers. They are epimers. They are epimers. Okay? What are they? They are epimers. Okay? What are they, students? They are epimers. Chale, next. Now, glucose. Now, glucose. Glucose. Okay, when you talk about the glucose, so you know that this glucose is what? Right? You know that this glucose is what? It is our, yes, main respiratory substrate. It is our main respiratory substrate, yes or no? It is our main respiratory substrate, yes or no? Yes or no? Yes or no? है ना? And this glucose is also known as blood sugar. Right? Usually in the fruits you have the fructose, but in the grapes you will see that glucose is present. So it is also known as the grape sugar. It is also known as the grape sugar. So it is the blood sugar, main respiratory substrate it is. It is the blood sugar, main respiratory substrate it is. Right, bache? So it is one of the most abundant organic compounds on earth. Okay, instant source of energy it is. Right? Right? Okay, so fructose, you know what fructose is? Fructose is known as fruit sugar. It is the sweetest sugar, right? It is the sweetest sugar, sweetest carbohydrate. Yes, bache? Yes, bache? It is the sweetest carbohydrate. And let me tell you, so it is the fruit sugar because it is there in the fruits, but in the glucose, in the grapes, you will see your glucose is there. And moreover, bache, when you talk about the fructose, even in honey, this fructose is present. Now, when you talk about the galactose, do you know galactose is the one that is known as brain sugar? Do you know galactose is the one that is known as brain sugar? It is the galactose that is the brain sugar. Brain sugar, right? It doesn't mean that your brain cells are going to feed on this galactose. No, it's not like that, right? Actually, it is the most abundant in brain and nervous tissue. That's why you call it as brain sugar. It is most abundant in nervous tissue, yeah, brain tissue. That's why. And it is not there in the free form. Freely it is present there, and these cells are the cells here are the brain and nervous tissue are feeding on it. No, it never occurs in free form, but it never occurs in free form. Matlab it is not present alone, but always matlab it's the part, it is making that tissue, it is involved there. So it occurs but always occur as a what? As a component. Right, bache? Right, bache? Right? As a component. Just I'll give you the example. And yes, let me tell you, this galactose na, it is most readily transported, absorbed. This galactose is most readily transported. Now I said it is the component. Just say lactose. Lactose is made up of, it's a type of oligosaccharide. It is made up of glucose and galactose. So in the brain and the nervous tissue, that's how your galactose is present. It's not like that, and not just in the, and yes, that's how your galactose is present. That's how your galactose is present, right? So it is there in lactose, it is there in agar-agar, it is there in some glycoproteins, right? So it is not used by brain cell, it is just the part of their structure, right? It is not used by brain cells. Brain cells are not using it, but it is just the part of their structure. It is just the part of their structure. Okay, it is just the part of their structure. Clear? Clear? So that is your, yes, monosaccharides. So now we will understand the oligosaccharides, but before that let's have a break. So it's 9:30, right? See you all at 9:45 p.m. right? We started the session late, so you will not get breaks for half an hour or something. Okay, so see you all at 9:45. Done? See you all at 9:45. Hi students. Hi students, so ready to start it? Yes? Am I audible? Am I audible? Okay. So now let's discuss the oligosaccharides. Okay. Now what we need to discuss? We will talk about oligosaccharides. So oligosaccharides, that's what we need to discuss. So I told you already in the these oligosaccharides, they are formed by the condensation, right? By the condensation of 2 to 10 monosaccharides, right? It is formed by the condensation of what? Of 2 to 10 monosaccharides. That is what we have. Yes. That is what we have. Tell me quickly. They are formed by the condensation of what? By the condensation of 2 to 10 monosaccharides. Okay? So, it's simple. When you join two monosaccharides together, what will you get? You will get disaccharide. That's the part of your syllabus. So, disaccharide is also a kind of oligosaccharide. When three monosaccharides are attached to each other, right, then you have what? Then you have trisaccharides. Then again, trisaccharides are also what? They are also the oligosaccharides, so etc, etc. So basically when 2 to 10 monosaccharides they join, they are going to give us what? They'll come under the category of oligosaccharides. Okay? So this disaccharide topic is important for us. Okay, so here you will talk about the lactose, you will talk about the maltose, you will talk about the sucrose, right? That's what we need to discuss. This is the part of our syllabus. Basically we need to know it. Now digestion and absorption is deleted from our syllabus, but before that before this revised syllabus now, in the digestion and absorption, when we used to discuss digestion of carbohydrates, there we used to discuss that. Okay, so still you people should know this. So the main question here is that how these monosaccharides they will join. How these monosaccharides they will join? How will they get connected to each other? Right? How will they get connected to each other? So obviously it is the glycosidic bond. Yes, obviously it is the, which bond is there? Question can directly come from the bond part. It is the glycosidic bond. Which bond is there, students? It is the glycosidic bond that is going to link these two monosaccharides together. That is going to do what? That is going to link these two monosaccharides together. Okay, so let's talk about this bond, right? Let's discuss about this bond. Which bond is there? Glycosidic bond, right? It is the glycosidic bond. Is that clear? Is that clear? Yes. Is that clear? Okay, so when you talk about the glycosidic bond, what's going to happen? Obviously, there is the minus H2O will be there. So, dehydration will be there. Dehydration will be there. There will be minus H2O. Now, let me explain this to you. See, you have one glucose molecule. Look at this. H, OH, H, OH, OH, H, H, OH, H, CH2OH, right? So let's take the example of maltose. Bache, maltose means 2 glucose, means glucose plus glucose is your maltose. When glucose is linked with glucose, you will get maltose, malt sugar, right? It is the malt sugar. So you have 2 glucose molecules here. Yes, you have 2 glucose molecules here. Can you see that? What do you have here? You have 2 glucose molecules here. Yes. Yes. So basically, this is the first glucose. Here you have carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. And here also you have carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 and carbon number 6. and carbon number 6. Now what is the point here? Here, the first glucose, it's carbon 1, and the second glucose, it's carbon 4. Here, right, minus, it will be dehydration, minus H2O, right? Here, the H2O molecule will be removed. So if the H2O will be removed, what will you get? See this. So in between carbon 1 and 4. So this, here you have H, here you have H, and it will join because water is removed. Water is removed. That's how it will link. So it is your glycosidic bond. This bond is your glycosidic bond. Simply, that's how we write it now. That's how we write it now. HO. 1 minute. HO. That's how we write it. Am I clear? That's how we write it. Okay, bache, it is the glycosidic bond. Which bond it is? It is the glycosidic bond. So basically, it is the bond which is in between first carbon and the fourth carbon of next sugar. So it's 1,4 glycosidic linkage. What's that? It's 1,4 glycosidic linkage. What is that? It is 1,4 glycosidic linkage. So you can see this here. So you have glucose, glucose. Obviously, these two are the alpha, right? Basically, this is the these are the two monosaccharides. Okay, bache? Right? Two monosaccharides are there. So basically, here the bond formation is in between first and fourth carbon. So that's how the bond will form. It is the disaccharide, it is the maltose. It is a disaccharide, it is the maltose, also known as malt sugar, right, bache? Also known as malt sugar, maltose, also known as malt sugar, right? Right? And here, even here, it should be HOH. I don't know why I'm writing like this. HOH. Okay? Malt sugar. Okay? So condensation will be there. Condensation will be there. Dehydration will be there. And this is what you people are going to get. So maltose is the malt sugar in the germinating seeds. We are going to see this. Right? This is the malt sugar. Okay? Yes? Okay? Right? So 1,4 glycosidic linkage is there. Ye dekho, maltose. That's your maltose. Okay. So now I told you about 3, right? I told you that we are going to discuss about these 3 disaccharides, right? Lactose, maltose, and sucrose. Lactose, maltose, and sucrose. Lactose, maltose, and sucrose. Just write it like this: maltose, lactose, sucrose. You can write it in any way. So malt sugar made up of glucose, 2 glucose subunits. Can I say that it is glucose plus glucose? Yes, students. Yes, bache. Can I say that it is glucose plus glucose? Yes, ma'am. Now it is the lactose. So it is glucose plus lactose. Lactose. So remember galactose. Remember galactose. Then sucrose. So it is glucose plus fructose. It is glucose plus fructose. Okay? So we have maltose, glucose plus glucose, lactose, glucose plus galactose, sucrose, glucose plus fructose it is. Glucose plus fructose it is. So here it is the sucrose which is the non-reducing sugar. It's a very important point. We will discuss it. It is the non-reducing sugar. These two are the reducing sugars only. These two are the reducing sugars only. So ye dekho. This is your lactose. So in the case of lactose, here you have beta galactose. Here you have beta galactose. The structure is very simple. The structure is very simple. You know that galactose is the epimer. Galactose is the epimer of glucose. Do you remember that? At carbon number 4, the H and the OH will be interchanged. At carbon number 4, the H and the OH will be interchanged. At carbon number 4, the H and the OH will be interchanged. Understood? Yes? 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 Right? So, it's beta galactose. So, if it is galactose, that is how it is going to be. So please focus on the structure, because it is what? It is beta galactose. What's that? It is galactose, beta galactose, beta galactose. So it's first carbon, second carbon, third carbon, and look at the fourth carbon. So that's how the OH and H is going to be. Now galactose plus glucose it is. This is your glucose. Understood? This is your glucose. Any doubt? This is your glucose. Yes? This is your glucose, and when it is the lactose formation, even your glucose is also beta glucose. It is both are beta. Both are beta. So obviously the bond formation will be there. The bond formation will be there. So there will be minus H2O. There will be minus H2O. So when you draw the bond here, na, even if you will see the book, so it is like this. Yes. Have you seen that? It is like this. It is like this. So it is your, again, it is your glycosidic bond only. It is your glycosidic bond only, and it is your 1,4 glycosidic bond only. But here you don't have the alpha sugar, you have the beta sugar. So sometimes it is mentioned as beta 1,4 glycosidic linkage. And this type of linkage you will see in the cellulose, which is a polysaccharide. Beta 1,4 linkage is there in the cellulose also. We will discuss about it. It will be there in the cellulose also. We will discuss about it. Okay, ji? So it is your lactose. It is your lactose. Okay. Okay. Next is your sucrose. Next is your sucrose. So when it is sucrose, sucrose is our normal table sugar, the cane sugar that we eat. It is known as table sugar. It is known as cane sugar, right? It is also known as invert sugar. And why do we call it as invert sugar? That's what you need to answer. The hint is related to the rotation of plane polarized light. Right? Hint is related to the rotation of plane polarized light. So what's the word? The word is sucrose. So it is made up of what? Glucose plus fructose. And what I just said, that it is the non-reducing sugar. I will explain it. I will explain it. Have patience. So glucose, alpha glucose, but the fructose is beta. Now fructose is also a hexose, but it is what? It's a ketose. It's a ketose. So how to draw its structure? Fructose. The structure of fructose is important. CH2OH, OH, HOH. Yes, am I right or wrong? Yes, the structure of fructose, is it correct or not? Please tell me, even I don't know, you tell me. The structure of fructose is correct or not? Yes, students, the structure of fructose is correct or not? The structure of fructose is correct or not? CH2OH, OH. Yes, please. Is it correct or not? No, ma'am, it's not correct. Why? Ye dekho, first carbon, second carbon, then it is the third one, no? It is the third one, na? It is the third one, fourth one, and then fifth one, and the sixth one. So it is like this, right? So the structure of fructose is important. Okay, the structure of fructose is important. Clear? The structure of fructose is important. Still, if there is any mistake, let me check if there is any mistake or not. Hanji, if there is any mistake, do let me know. First, second, third carbons. Fructose. So here the ring is different. So according to you, what should be the structure? According to you, what should be the structure? You're talking about the fructose. Yes, you're talking about the fructose here. First carbon, second carbon, third carbon will be OHH, fourth carbon will be HOH, fifth carbon. So this is correct. Okay. So here this one is our first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and this one is your sixth carbon. Okay. So it will be at first carbon CH2OH and then H. So it's a furanose, right? It's a furanose. Okay. What's that? It is a furanose. Okay. So it is basically your, yes, it is basically your alpha fructose, right? Alpha fructose, because at first carbon, right? Okay. Yes. So if it is your alpha, fructose, because now you don't have HOH. At first carbon, basically this is the first carbon. So here you have CH2OH. Here you have CH2OH. So it is alpha fructose only, right? It is alpha fructose only. Or you can say that it is alpha fructofuranose. It is alpha fructofuranose. Okay, so this is the correct structure. This is the correct structure. So hydroxyl group at C1 is situated in the opposite direction to the CH2OH at the sixth carbon. है ना? At the sixth carbon, ये देखो, this is the opposite direction. So this is your alpha fructose. So when you look at the structure, ये देखो, right? So this is your beta D fructose. This is your beta D. So if my alpha fructose is like this, so when you talk about the beta, beta fructose, then it will be OH, sorry, OH, CH2OH, OH, H, OH, and this one is same. So this one will be your beta. This one will be your beta, right? So the fructose, this is your beta fructose. Okay, this is your beta fructose. And do not get confused, because sometimes now, if you see it on Google or somewhere, just they write it like this. They consider this side as the first one. And even we should consider it like this, because I have to show you the bond formation. That's why I'm drawing it in such a way. So it will be your CH2OH, OH, H, OH, H, OH, CH2OH, it will be then. That's how it is, right? So in some books it's like this: 1, 2, 3, 4. Sorry, 1, 2, 3, 4, 5, and this one is the sixth carbon. So don't get confused. Okay, please don't get confused. Okay. So in some books it is like this. They have started the numbering from this side, and here I'm showing you it in this way. So both are correct. So this one is alpha, right? And here you need beta. Here you need beta. So OH should be somewhere here and CH2OH should come down. Okay? Beta. Okay? Okay. So here also the linkage will be there, and that will be your beta. Right, the glucose is alpha, but the fructose is beta. But that much detail is not required. So simply you can write that here, when it comes to the bond formation in sucrose, it's 1,2 glycosidic bond. It's 1,2 glycosidic bond. Why 1,2? Because the first carbon of your glucose, but the second carbon of your fructose. Please remember it. First carbon of your glucose, second carbon of your fructose. So in the polysaccharides also, just say you talk about the polysaccharides, we discuss one example, inulin. Inulin is a polymer of fructose. Inulin is a polymer of fructose. So definitely glycosidic bond will link. But when there is the fructose involvement, the first carbon of glucose, and even if there will be fructose, first carbon of fructose, second carbon of next fructose. So it will be 1,2 glycosidic bond, not 1,4 glycosidic bond like your lactose and maltose. Okay? Like your lactose and maltose. Okay? Okay, so it is commercial sugar, alpha D glucose and beta D fructose is there. ठीक है? So I told you here about the structure also, that what are the changes you need to take care. Okay, what are the changes you need to take care? जैसे ये देखो, this is your fructose, CH2OH, HOH. So sorry, this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. That's it. Okay, that's it. So glyceraldehyde, ribose, deoxyribose. See, this is your mannose. I told you already, they are epimers. So at second carbon, H and OH is interchanged. Rest everything is same here, right? Rest everything is same here. So accordingly, the structure of glucose in the ring form will be. That's it. That's it. Okay. So this is about the disaccharides, and I told you that sucrose is nothing but it is the invert sugar, table sugar, commercial sugar, cane sugar, and most important, it is also the non-reducing one. It is also the non-reducing one, right? It is a non-reducing sugar. Yes or no? Yes or no? Please tell me yes or no. Right, it's a non-reducing sugar. So it cannot reduce your cuprous to cupric या cupric to cuprous. Cupric to cuprous या cuprous to cupric. Please tell me. Cu2 positive, what's that? Cu2 positive, what's that? Is it cuprous or is it cupric? Cu2 positive. What is it? Cuprous or cupric? Cuprous or cupric? Cu2 positive. बच्चे, Shivam, are you sure cuprous to cupric या cupric to cuprous? It is cupric to cuprous. Cu2 positive is cupric, na? नहीं? Cu2 positive is not cupric. Is it cuprous? Komal, are you sure? Komal, are you sure? Guys, start revising. Cu2 positive is cupric, not cuprous. Not cuprous. Yes. Please do not make mistake here. So Cu2 positive is cupric, and then cuprous, right? So it will not be able to reduce cupric to cuprous. It's the non-reducing sugar. It's the non-reducing sugar, okay? It's the non-reducing sugar. I'll tell you its function as well. Any doubt? Any doubt? Any doubt? Pakka? Sure? Chaliye. So now we need to discuss polysaccharides, okay? We need to discuss polysaccharides. Ready? What are we going to discuss now? Now we will discuss polysaccharides. Polysaccharides. So what do you know about the polysaccharides? What do you know about the polysaccharides? Yes, what do you know about the polysaccharides? What do you know about the polysaccharides? Yes. Obviously, they are the long chains of sugars, right? And of course, here you will see more than 10 monosaccharides, right? It is never be less than monosaccharide, but more than 10 monosaccharides. What are you talking? You are talking about the polysaccharides, right? What are you talking? You are talking about the polysaccharides, right, bache? So when you talk about them, their suffix is n. जैसे कि glycan, glycans, ns, n. So suffix, the one that will come at end, it is an. It is an, right, bache? It is an. Yes or no? Yes or no? So when you talk about these polysaccharides, na, obviously they are made up of many monosaccharides, and when you talk about their chemical properties, they are non-sweet. They are not sweet, right? So you cannot say that all the carbohydrates are sweet. No, they are not. They are tasteless. They are tasteless and they are insoluble in water. They are not even soluble in water. They are insoluble in water. And bache, they are non-reducing in nature. Right, they are non-reducing in nature. Just I told you, monosaccharides all are reducing in nature. But here in polysaccharides, they are non-reducing in nature. And yes, there is a line of NCERT that because there will be two ends, na, right end of the chain and left end of the chain of the polysaccharide chain, because it's a polymer. Just let's say so many subunits, they join together, right? So one will be the right end, one will be the left end. So right, R for right, R for right, R for reducing, R for right, R for reducing, and left end is non-reducing. So this line is there in the NCERT, and in many questions of this, many times you will see it as a PYQ, right? And many times you will see it as a PYQ, right, bache? Right, bache? Sure. So what's the word that we have here? That they are the long chains of sugar, right? We are calling it as glycans. They are non-sweet or tasteless, right? They are insoluble in water. They are non-reducing in nature, right? They are non-reducing in nature. So right end is reducing, left end is non-reducing one. So overall they are non-reducing, but if you specifically talk about right end and the left end, that's what you need to mention. Okay, that's what you need to mention. So right end is called the reducing end and the left end is called the non-reducing end. Left end is called the non-reducing end. Okay. Okay. So obviously as per their function, they can be nutritive or they can be structural polysaccharide. Matlab they are meant for storage and even for the nutrition. Just say when it comes to the storage polysaccharide, can you quote any example? Yes. Can you quote any example? Storage polysaccharide or structural polysaccharide? Storage polysaccharide or structural polysaccharide. It is as per their function. Storage polysaccharides and structural polysaccharides. So structural means it helps in the formation of structural, and storage, जैसे कि your glycogen, it's a polysaccharide and it is our stored food. Animal starch it is, right? Your starch itself. Okay, even the inulin, it's not insulin. Insulin is a protein, it is inulin. This question has been asked in NEET 2020, inulin and insulin. Insulin is a protein, inulin is a carbohydrate. It's a polysaccharide. It's a polymer of fructose. Now structural mein, jaise you have cellulose, plant cell wall, you have chitin, etc. So that's what you need to keep in your mind. That's what you need to keep in your mind. So that's how we divide. And the other thing is, now it's a polysaccharide, right? Means it is made up of many monosaccharides. So when you talk about the polysaccharides, that's how we divide it. On the basis of their monomers, right? On the basis of their monomers also we divide it. Okay? Okay, just let's say if they have same type of monomers. You have two words here. One is homopolysaccharides. One is homopolysaccharides, another one is heteropolysaccharides. One is homopolysaccharide, another word is heteropolysaccharides. So when it is homo means same. Homo means same type of monomers, same type of monosaccharides are making these type of polysaccharides. ठीक है बच्चे? Yes. Same type of monosaccharides are making this type of polysaccharides. When it is hetero, different types of monosaccharides are there, right? Different types of monosaccharides are there, right bache? Different types of monosaccharides are making the structure here, heteropolysaccharide. So homo means same type of monosaccharide, hetero means different. So when it is the homopolysaccharide, you have the example of glycogen, right? You have the example of starch, you have the example of cellulose, you have the example of inulin, you have the example of chitin, right? Right, etc, 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 etc. Yes, yes. And when you talk about the heteropolysaccharides, you have the example of hyaluronic acid. I'll discuss it. You remember hyaluronic acid, cementing material it is. Yes. Do you know about hyaluronic acid? Anybody here in the class, do you know about hyaluronic acid? Hyaluronic acid, it is the cementing material. We discuss it in extracellular matrix. It helps in that connection. So it is the cementing material. Hyaluronic acid, right? Other than hyaluronic acid, heparin, which is the natural anti-blood coagulant, right? Hyaluronic acid, heparin, which is the natural anti-blood coagulant. Other than that, your hemicellulose, that is present in our plant cell wall. Hemicellulose, your pectin, your chondroitin. So they all are the example of heteropolysaccharides because they are not made up of same units, right? They are not made up of same units, right bache? Right bache? So the first one is the cellulose. So you know that cellulose, the structural polysaccharide, right? It is there in the plant cell wall. It is there in the plant cell wall. So bache, it is linear. Linear, yeah, you can say that unbranched straight chain it is. When I'm saying linear, man is straight chain, na? So it is linear, unbranched. And as I said, it is the example of homopolysaccharide. As I said, it is the example of homopolysaccharide. So basically your beta glucose, beta D glucose to be more specific, right? So many beta glucose, many beta glucose, many beta glucose, many beta glucose will together form your cellulose. So obviously there will be the glycosidic linkage, and that is your beta 1,4 glycosidic linkage. That is your beta 1,4 glycosidic linkage. That is your beta 1,4 glycosidic linkage. Understood? Understood? Beta 1,4 glycosidic linkage, cellulose. After cellulose, I'll give you the example of inulin here, right? I'll give you the example of inulin here. What's the word? Inulin. So, bache, it's a polymer of fructose, right? Right? Yes, bache, it's a polymer of fructose. What is it? It is a polymer of fructose, right? And this is because it's a polymer of fructose. So glycosidic bond is there, right? Because it is the glycosidic bond that links the various monosaccharides. But here you will see 1,2 glycosidic linkage, right? Here you will see 1,2. Matlab first carbon of the first sugar, second carbon of the next sugar is involved in bond formation. Yes, it is involved in bond formation. It is involved in bond formation, right, bache? Right, and definitely it is beta 1,2. Then this inulin is found stored in the roots of, there is a plant, and this plant even propagate by the roots of dahlia and artichoke. And this is used to, this inulin is used to check GFR. So what is GFR? Glomerular filtration rate it is. It is glomerular filtration rate. What is this GFR? It is glomerular filtration rate. What's that? It is glomerular filtration rate. So it is used to know or check the GFR. Okay? Okay? Yes? Right? Understood? So cellulose, inulin. Next. Chitin. Very important it is, chitin. So bache, chitin is also a polymer. It's a homopolysaccharide, but it's a polymer of N-acetyl glucosamine. N-acetyl glucosamine, that is NAG. Now see, Homo means same type of polysaccharide. Just remember, if it is a circle, all are going to be circle. All are going to be circle, right? If it is a circle, all are going to be circle. So that's why we are saying that it is what? It's a homopolysaccharide. That's why we are saying that it is what? It's a homopolysaccharide. Okay? That's the first point. Now, what is my second point here? This is glucosamine. Matlab, this is a modified monosaccharide, you can say so. But chitin is made up of so many nags. So many nags together are going to make the chitin. Okay? So many nags together are going to make the chitin. Is that clear? So many nag together are going to make what? They are going to make the chitin. Right, bache? They are going to make the chitin. Right? So many nag are going to make this chitin. ठीक है बच्चे? So it is also linear, or you can say that unbranched straight chain. Unbranched straight chain. ठीक है? And we know that this chitin is very important because it is also known as fungal cellulose. It is present in the cell wall of fungus. So it is also the fungal cellulose. It is present in the cell wall of fungus. This chitin plus. It is also there in the exoskeleton of insect, a very important topic, right? Exoskeleton of insects will also contain this chitin. So ultimately beta glycosidic linkage is going to keep it all together, right? And here you have 1,4 linkage. Here you have 1,4 linkage, right? Beta 1,4 linkage again. Here you have beta 1,4 linkage. Okay? Now next that we need to discuss is glycogen and starch. Let me tell you, they both are branched. They both are branched polysaccharides. They have the branching as well. They have the branching as well. They both are branched polysaccharide. So understand this concept of branching. And because they are ultimately the homopolysaccharides, so ultimately they are made up of glucose only. Ultimately they are made up of glucose only. है ना कोमल? Ultimately it is made up of glucose only. They are the branched polysaccharide, but ultimately they are made up of what? They are made up of glucose only. Understood? Done? Sure? Right? So understand this branching point. Branching point. जैसे ये देखो. Yes, bache. So that's what you have. That's what you have, so many glucose. So obviously you know that the glycosidic bond will form here. The glycosidic bond will form here. We call it as 1,4 glycosidic linkage. Right? What's that? It is 1,4 glycosidic linkage. What's that? It's 1,4 glycosidic linkage. Am I right? Am I right? Here also, what will you see? Here also, what will you see? 1,4 glycosidic linkage. 1,4 glycosidic linkage. Now imagine it in this way. Just say you have this straight chain. Now at the point of branching, just say, let's say here you have the branching. Then again rest all will be in the straight chain. Right? Yeah, this point is also of the branching. Then again they all will be in a straight chain. Right? Are you getting my point? Are you getting my point? So alpha 1,4 linkage when there is a straight chain. 1,2 linkage when there is the fructose. When there is the fructose, 1,2 linkage will be there. I told you that. And other things are also there when, chalo, let's not enter there. Now at the point of branching, just say ye dekho, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6th. This is the 6th carbon. Now let's say here you have the glucose. This is the point of branching. This is the point of branching. HOH is there. HOH, HCH2OH. So at the point of branching, the sixth carbon here and the first carbon here, they are involved in glycosidic bond formation. They are involved in glycosidic bond formation. So glycosidic bond will form in between the sixth carbon of this sugar and the first carbon of the next. So wherever you will see branching point, the bond will be 1,6 glycosidic bond. So if 1,6 glycosidic bond is given, means it's the point where the branches will form. Where the branches will form. Is that clear? Is that clear? Is that clear? Done? Done? Yes, done? So at the point of branching, you will see the 1,6 bonding will be there. जैसे ये देखो glycogen. I'll discuss it in detail. See the straight chain 1,4. The straight chain 1,4. Wherever there is the branching, sixth carbon, and this is the first carbon. So there will be 1,6 linkage. There will be 1,6 linkage. There will be 1,6 linkage. So it is the glycogen, animal starch. It is bache. Animal starch, it is the stored form of food in our case, right bache? Yes? Done? 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 So it is multi-branched. Many branches are there. So it is multi-branched. You know that it is stored. It is also known as the animal starch. Even in the case of fungi, the glycogen is the stored food. Even in the case of fungi, glycogen is the stored food. So it is the multi-branched. It is multi-branched. Told you about the bonding as well. Right, bache, also known as animal starch. And it gives red color with iodine. It is not going to give you the blue color. Blue color we will see in the case of your starch. Okay, so it gives, and to be more precise, amylose. So it gives red color with iodine, right? It gives red color with iodine. What will it give? It will give red colored with iodine. It is multi-branched, students. It is multi-branched, and yes, it is stored where? Where is it stored? In the liver and the muscles. Okay, it is stored in liver and muscles, and you know that, right? Majorly in the livers. Matlab if I have to compare, so obviously in the liver it is stored, right? Matlab more quantity is there in the liver. And bache, if we need to check the number of if we need to check the ends here, matlab there is a formula, number of ends in glycogen, right? There is a formula there to check the number of ends. So if we have to check the number of ends in glycogen, so it will be 1 plus number of branches. Just if 10 branches are there, 10 plus 1, 11 ends will be there, right? So 1 plus number of branches. 1 plus number of branches. 1 plus number of branches. Understood? 1 plus number of branches. Understood? Pakka? Yes? ठीक है. So ये देखो glycogen. Look at this. It is multi-branched. It is highly branched. So number of ends is equals to 1 plus number of branches. Number of ends is equals to 1 plus number of branches. Understood? 1 plus number of branches. ठीक है? Right? चलिए. Next is your starch. stored food in the case of plants. Stored food in the case of plants. And let me tell you, in the case of plants, you know that phloem is present. Phloem is present. Xylem and phloem is there for the translocation. Xylem and phloem is there for the translocation. Translocation means when we have to transport something at a sorry at a longer, right? When there is a small distance transport, then diffusion and osmosis is there. When there is the long distance transport, xylem and phloems, they are involved. Xylem and phloem is involved. What is involved? Xylem and phloem is involved. So when xylem and phloem is involved, bache, when xylem and phloem is involved, when what is involved? When your xylem and the phloem is involved, so we will call it as translocation. We will call it as translocation. We will call it as translocation. So in the phloem, it is the sucrose that translocate, the non-reducing sugar. And ultimately the stored food in the case of plant is starch. So starch is also a homopolysaccharide indeed. It is also a homopolysaccharide indeed, right? But what do you see here? Yes, what do you see here? This starch is also what? It is homopolysaccharide. So what do you see here? It's simple. Starch is having amylose and amylopectin. Now sometimes some students, they ask me, ma'am, if amylose and amylopectin is there, how can we say that it is the homopolysaccharide? Arey yaar, amylose and amylopectin is made up of glucose only. It is also made up of glucose only. So amylose is basically the linear, matlab it is unbranched. Sorry, it is unbranched but helically coiled. It is unbranched but helically coiled, right? This one is branched. Amylopectin is branched. So what is the meaning of being unbranched but helically coiled? Simple. So let's say this is the straight chain. This is the straight chain, right? Your amylose is like this, but it is helically coiled. It is unbranched. It doesn't have branches, but it is helically coiled. So because it is helically coiled, so it will trap the iodine, right? It will basically hold the iodine and it will give blue color with iodine. Okay? So amylose, it's linear. So obviously linkage will be 1,4 only, and it gives blue color with iodine. That's why starch, whenever we need to check the presence of starch, we test it with the iodine. If blue color is there, starch is present. And why that blue color? Because amylose, because of amylose, because it will hold it, okay. Okay, so it is branched. So if it is branched, the bonding will be alpha 1,4 and 1,6, and it gives, matlab individually if you will check amylopectin, it gives red color with iodine only, right? Red color with iodine only. Understood? So amylose is basically 20 to 30%. 20 to 30%, and amylopectin is like this. Okay, so the ratio will be Ambika. The ratio will be 1/4. 1 part of amylose, 4 parts of amylopectin. That is our starch. Okay, that is the starch. Okay. Yes, bache, is it okay? So this is amylose, amylopectin 1,6 and 1,4, and amylose is having 1,4. Amylose is having 1,4. Amylose is having 1,4. Done? 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 Okay, amylose and amylopectin. So amylose may only 1,4, here 1,4, but branches is there, so 1,6 linkage. So linkage part clear? Linkage part clear? Yes, that's your amylopectin. That's your amylopectin, okay? So this is all about the polysaccharides. Baki, I told you, you have the heteropolysaccharides as well, right? The hyaluronic acid is there, right? Heparin, hemicellulose, pectin, and right, and chondroitin is there. Okay, so pectin is considered as plant cement. Okay, pectin is considered as plant cement, and hyaluronic acid is also the cementing material, but this is the animal cement, right? You will see it in the animals. So it is considered as animal cement, hyaluronic acid, and P for pectin, plant cement. Okay, so it is present in the plant cell wall, and you know that it helps that cell to fight with the pathogen as well, to fight with the infection as well. Okay, chaliye, so that's it. And you also have mucopolysaccharides, right? Mucopolysaccharides. So mucopolysaccharides are the one that can form slime and mucilage, right? They can form slime and mucilage. Mucopolysaccharide is the word. These are slime and mucilage producing polysaccharides. These are slime and mucilage producing mucilage producing polysaccharides. Okay, slime and mucilage producing polysaccharides. What are they? They are the, yes bache, they are the mucopolysaccharides. Okay, so basically they have the capacity to bind proteins and water, right? Because plants mein to mucilage is very common. So they have the capacity to bind with proteins and water. So you have example, jaise agar-agar. It is an example of what? It is an example of mucopolysaccharide. Right, bache? Right, bache? And other than agar, your peptidoglycans, they are also the example of your mucopolysaccharides, okay? They are also the examples of your mucopolysaccharide, okay? So bonding part clear, polysaccharides clear. Next part is easy. Next part is easy. Next part is easy. Okay, so in the NCERT, yeah, yes, so see this. In the NCERT also it is given, and these are the general things regarding lipids and all. So in the NCERT even this table is given na. So here what are they showing? Average composition of cell. Maximum is water. After water it will be protein. After protein it is going to be nucleic acid, then carbohydrate, then lipids, and then iron. You should know about it. So maximum is water, then proteins, then nucleic acid, then carbs, then lipids, and then ions. Right, then ions, okay, then ions, okay, yes. So polysaccharides, ye dekho, cellulose, starch, all clear. So that's the diagram. This is the representation of portion of glycogen. So how to check the number of ends? 1 plus number of branches. How to check the number of ends? 1 plus number of branches, right? 1 plus number of branches. So here dekho, plant cell wall is made up of cellulose, paper made from plant pulp and cotton fiber is cellulosic, right? So they have, okay, so there are more complex polysaccharides in nature, right? They are the building blocks, amino sugars and chemically modified sugars, like glucosamine, N-acetylglucosamine. So you know about the chitin, one of the most important examples. So that's it. So the next thing that we are going to discuss is your lipids. Okay, let's talk about the lipids. So any doubt? Any doubt? Yes, any doubt? So, bache, to understand polysaccharide, you should know about the monosaccharide, oligosaccharide and the bonding part, right? You should know about it. So, now we will talk about lipids. Lipids. Now we will talk about lipids. Okay. Yes. Lipids. Micromolecules they are, right? But where are they present? But present in acid insoluble fraction. But where are they present? They are present in acid insoluble fraction. Acid insoluble fraction. But still, what are they? They are not strictly macromolecules, they are micromolecules. So in the case of lipids, remember their molecular weight is 18 to 800 Daltons. It lies in 18 to within this range, 18 to 800 Daltons, right? Yes. Okay. So when you are saying lipid, so what are lipids? Fats, obviously, and its derivatives. Fat and its derivatives, they are known as lipids. I'll discuss about the types of lipids also. Right, I will discuss about the types of lipids also. So obviously here also these are the compounds having, these are the compounds, yes, even these are the compounds having carbon, hydrogen and oxygen. But in the carbohydrates, hydrogen and oxygen is in the ratio 2:1. But in the carbohydrates, hydrogen and oxygen is in the ratio 2:1. But here. So these are also the compounds having carbon, hydrogen and oxygen. But hydrogen and oxygen is not in. But hydrogen and oxygen is not in 2:1 ratio. That's what you have to remember, right? That's what you have to remember. Right, and that is the reason, because hydrogen and oxygen is not in 2:1 reason. So that is why it needs more oxygen. It needs more oxygen for oxidation. We say that carbohydrates, right? We say the carbohydrates, the glucose is the main respiratory substrate. When our body needs the food, needs the energy immediately, so we are dependent on the glucose, right? We are dependent on what? We are dependent on the glucose. Yes or no? We are dependent on what? Anji, Ramesh? Yes. We are dependent on what? We are dependent on glucose. Yes or no? Yes or no? Yes or no? Yes, definitely. Yes. So why? But overall, the fats, they give more energy, but they are not our main respiratory substrate. We cannot rely on them, right? We cannot rely on them for the immediate energy requirements. Why? Because it takes a lot of energy for oxidation, right? It takes a lot of, it takes more oxygen for oxidation, and even you know that their absorption is not as simple, right? Their absorption is not that simple. Okay, their absorption is not that simple. So lipids, molecular weight is 18 to 800 Daltons. Lipids are actually the fats and their derivatives. So they are the compounds having carbon, hydrogen, oxygen. But hydrogen and oxygen is not in 2:1 ratio. Hydrogen and oxygen is not in 2:1 ratio. Hydrogen and oxygen is not in 2:1 ratio. Understood? Understood? Yes. So because amount of oxygen is considerably very less here, right? Right. So lipids being insoluble in water, They are insoluble in water. I'm talking about their properties, but soluble in organic solvents. है ना? But soluble in organic solvents. But they are soluble in organic solvents. For an example, acetone, benzene, chloroform, etc. etc. Clear? Etc. etc. Right, bache? Yes. Yes. So fats, they are very important for our body, right? Fats, they are very important for our body. Yes or no? Are they important? Yes or no? Yes. So you know that they are present in our subcutaneous layer, right? They act like a reservoir. They act like a shock absorber. Just say if you talk about the kidney or any body organ, they are covered with the adipose tissue, right? It is acting like a shock absorber. It is acting like a reservoir also, right? It is also providing us the insulation, right? And when it comes to the lipids, right? They need less space for the storage in comparison to the carbohydrates. Lipid being insoluble in water, they can form even the vesicles, na, you know it already. So lipids, when you talk about the lipids, students, when you talk about the lipids, remember they need less space for the storage in comparison to the carbs. Right, bache? Right? So lipids, it is there in our subcutaneous layer. You know about that, right? You know these things about the lipids. Right, bache? Right? So maximum amount of food is stored in the form of lipids. So whatever we are going to take, just say, let's say, obviously our body needs carbs, protein, lipid, everything. But if let's say you are eating something in excess, you are eating excess of carbohydrates, excess of proteins, if there is anything that you are taking in excess, na, ultimately our body will convert it into the form of lipids, in the form of fats. Okay? In the form of fat. So whatever you are going to eat in excess, ultimately our body will convert it into the lipid. Let's say you are eating protein. Our body needs a certain amount of protein. Our body will use it. And then if there is any extra protein, it will be converted to the lipids. Right? Right. So that's what you need to know. That's what you need to know. So this is about the lipids. Okay. This is about the lipids. Some lipids can have P, N and S. Phosphorus, nitrogen and sulfur can also be there in some lipids, okay, in some lipids. So when you talk about the lipids, you need to know about this flow chart. Simple lipids, conjugate or compound lipids, simple lipids, conjugate or compound lipids, and derived lipids. ठीक है, simple or yes, simple lipids, conjugate or compound lipids and derived lipids. So when you talk about the simple lipids, see, in the lipids you will talk about the ester bonds, right? We will discuss it. So it is the esters, right? They are the esters of alcohol with fatty acids. They are the esters of alcohol with fatty acids. So when you talk about the simple lipids, here you will discuss neutral fats or waxes. Right? Neutral fats, yeah, true fats or waxes. Right? Or waxes. So as I said, esters of alcohol with fatty acid, I'll discuss that. So when they are the esters of glycerol, when the alcohol is glycerol, and for fatty acids I'm using the short form Fa, and when these are waxes, they are the esters of mono, mane single, hydric, OH, monohydric alcohol and fatty acids. Okay? Waxes are, right, waxes are esters of fatty acid with monohydric alcohol. Like bee wax is the example, or ear wax, also known as cerumen. They are the examples here. So what is the word here? They are the esters of alcohol with fatty acid. We are calling them as simple lipids. So when it is conjugate or compound, When it is conjugate or compound, so they are also the esters of alcohol with fatty acids, right? Obviously fatty acids will be there, that is for sure. So these are the esters of okay, the glucose is having the extra group, my bad. So they are the esters of fatty acids with alcohol containing other groups, जैसे your phospholipids, sphingolipids, like this. So they are the example of conjugate or compound lipids, right? So they have other groups, phospholipid, glycolipid, lipoproteins, like this, right? So derived lipids like your cholesterol, right? Your steroids, basically derived lipids. So they have that, right? Hydrocarbon chain is also there and the ring is also there. So when it is the derived lipid, so simple lipid mein to we have neutral fats or waxes. There you have phospholipid. A very common example is of phospholipid is lecithin. So derived lipids mein they have long hydrocarbon ring with hydrocarbon chain. Long hydrocarbon ring with hydrocarbon chain. Okay? Okay, so we will talk about the cholesterol. So is it clear? Then we will discuss simple lipids or neutral fats. So esters of long chain fatty acid and alcohol. Long chain fatty acid and alcohol. Done? Chaliye. So let's start with the simple lipids. Let's start with the simple lipids. So when you people talk about the simple lipids, bache, remember here these are, matlab majority of simple lipid are the esters of fatty acid and alcohol. And which alcohol it is? Majorly bache it is trihydroxypropane, or in simple words you can say it will be trihydroxy sugar alcohol. It will be trihydroxy sugar alcohol, right? Trihydroxypropane, I'm saying that like majority of simple lipids are going to have trihydroxypropane. That is your glycerol. What is your glycerol? I'll write it here. Glycerol, 3 carbon, 3 times OH, and rest CH2. Just satisfy the valency here. This is glycerol. This is your glycerol, right? That is your tri, means 3 hydroxy, 3 times OH, propane it is, because carbon number is 3, no? So trihydroxypropane. It is trihydroxypropane. So alcohol, right? Majorly, like in majority of simple lipids, you have the esters of fatty acid with alcohol. So that alcohol is trihydroxy sugar alcohol, and the very common one is the glycerol. Very common one is the glycerol. Clear? Right? Right? Very common one is the glycerol. Okay, ji? Now, fatty acids. Understand this concept. Right? When you are saying fatty acids, but it is having acidic group, right? Acid is the word here. Matlab they are having the acids. They are having the acid, yes or no? Right? Just we are simply saying that fatty acids, they will be esterified with alcohol. And alcohol, most commonly it is glycerol. So even glycerol is also referred to as the most simple lipid. Right? Even this glycerol is also referred to as the most simple lipid. Right? Right? So glycerol is also the simple lipid, trihydroxypropane. And many lipids have both glycerol as well as the fatty acids. And these fatty acids, they will be esterified with the glycerol. They will be esterified with the glycerol. Right, bache? Right, bache? Right? And when you talk about the lipids, na, just we say fats or oils. So it depends upon the melting point. On the basis of melting point, you categorize it as fats or oils. Right? You call them as fats or oils. They say oils will have low melting point. Right? Right? They will remain oil in the winter. They are having low melting point. Right? You can take the example of any oil. It will be oil in the winter. It is having the low melting point. Okay? It is having the low melting point. So now I'm talking about the glycerol. I told you it is the simple lipid. So some fats are also having the glycerol with the fatty acid. So this is the glycerol. Now the next word is fatty acid. Now the next word is fatty acid. Then I'll teach you what is the ester bond. Okay? Fatty acid. But it is acid. Definitely it will contain COOH bond, C double bond OOH. Right? Carboxylic group will be there for sure. Carboxylic group will be there for sure because it is an acid. Carboxylic group will be there. Yes or no? Yes or no? And this carboxylic group is attached. Matlab, if something is acidic, C double bond OOH should be there. And this is attached to the R group. Right? What am I saying here? I am saying fatty acid. I am saying that obviously it is an acidic structure. So definitely acidic group, carboxylic group will be there. So this carboxylic group is attached to the R group. Right? It is attached to the R group. So this R group could be methyl. So when I'm saying methyl, matlab CH3. Right? When I'm saying methyl, matlab CH3. It can be ethyl. That is C2H5. I will elaborate and explain it. Or higher number of CH2. Or higher number of CH2 groups. Matlab that CH2 can be from 1 to 19 carbons are there in the fatty acids. 1 to 19 carbons are there in the fatty acids. Just for an example, C double bond OOH. Methyl group means that's the condition. This is a C double bond OOH and this methyl group is attached here. That is also a fatty acid. Now let's say if it is C2H5 to this COH, that is how it is going to be C2H5. Now let's say, so now I just said that these CH2, so many CH2 groups can be there. So many CH2 groups can be there and it can be from carbon 1 to 19. Matlab, if carbon, only one carbon is there, matlab to this carboxylic group, CH3 is attached. If two carbons are attached to this carboxylic group means ethyl group is attached there. So it can be till 19th carbon, 1 to 19th. And obviously you will see C double bond OH. C double bond OH. Right? C double bond OOH. Right, bache? So CH2 groups can be from carbon 1 to carbon 19. Carbon 1 to carbon 19. Carbon 1 to carbon 19. So let me make it clear. In your book, there is an example that is arachidonic acid. That is arachidonic acid. So total number of carbon here. Total number of carbon here is 20. So some students are like, madam, you were saying carbon 1 to 19. How can it be 20? It's simple. Carbon number 1 to 19 in this CH2 group, in this R group. So this one will always be there, na? So if it is the arachidonic acid, it means this carboxylic group plus 19 other carbons will be there. So in totality, total number of carbon basically, in totality, is carbon is equals to 20. Right? C is equals to 20. Right? C is equals to 20. Okay? Okay? C is equals to 20. C is equals to 20. Okay? Yes, Samir. So fatty acid clear. Fatty acid clear. And glycerol clear. Now I'll give you the example. Just say glycerol 1. Now fatty acid. If it is a fatty acid, C double bond OOH group will be there. And let's say this is R1, this is the alkyl group. This is R1, this is the alkyl group. Okay? Now here, what is going to happen? See this carefully. What is going to happen? See this carefully. Here, this. Again, ester bond has to form minus H2O. Water will be released. That's how the esterification will be done. This is your glycerol. One glycerol, and here you have one fatty acid. So let's say you have 3 fatty acids here. But these 3 fatty acids can be same, they can be different. They can be same, they can be different. They can be same, they can be different. So glycerol is 1. Glycerol is 1. So water will be eliminated. Water will eliminate. So if water will eliminate here, water will eliminate here. So obviously, that's how it is going to be. That's how it is going to be. Right? That's how it is going to be. That's how it is going to be. So C double bond O, O C bond. Matlab when you are talking about the ester bond, it's just like this. It's just like this. Right? So this is your ester bond here. This is your ester bond here. Right? See this. You have glycerol. You have 3 fatty acids. So obviously they are going to form the bond here. Right? They are going to form the bond here. This is the ester bond. So you have 3 ester bonds here. So when one glycerol, when it attaches with 3 fatty acids, what will you get? Triglycerides. What will you get? Triglycerides. Triglyceride. Most common, matlab simplest lipid is your glycerol, but it is your most common type of lipid. Simplest is your glycerol, but it is your most common type of lipid. What's that? It is your most common type of lipid. Okay. What is it? It is your most common type of lipid. Doubt? Doubt? Yes. Do you have any doubt? It is the most common type of lipid. So, 1 glycerol, 2 fatty acids, diglyceride. 1 glycerol, 1 fatty acid, monoglyceride. So these fatty acids can be same, they can be different. Bond formation clear? Bond formation clear? Yes. Bond formation clear? Yes, please. Chali. So now, bache, when you talk about the fatty acids, you have 2 things here. Saturated fatty acid and unsaturated fatty acid. Saturated fatty acid and unsaturated fatty acids. So saturated, single bond. Unsaturated double bond can also be there. Right? Unsaturated double bond can also be there. Ye dekho. Firstly, see this. You have 1 glycerol, you have fatty acid. They can be same, they can be different. So COOH is there. Rest is carbon hydrogen chain. Okay. So condensation. Right? Condensation mein water molecule will be eliminated. That's how the bond will form. So this is triglyceride molecule. This is triglyceride molecule. Clear? This is triglyceride molecule. Yes? This is triglyceride molecule. Now, I'm talking about the saturated and unsaturated fatty acid. When it is saturated, carbon, hydrogen, no double bond. When it is unsaturated fatty acid, see, carbon, carbon double bond is there. That unsaturation can vary. Sometimes 1 double bond, sometimes 2, 3, it depends. Okay? So wherever there is the double bond, a kink, a kind of bend will be there. The same thing we discuss in the phospholipid bilayer, right? When we say saturated or unsaturated fatty acids are there. Okay, bache? Okay. Chalo. So now let's discuss it. So when it is the saturated fatty acid, what is the point? What is the point here? That no carbon to carbon double bond in the fatty acid. No carbon to carbon double bond in the fatty acid. No carbon to carbon double bond in the fatty acid. Right, bache? And here, one or more double bond. One or more double bond. Right? One or more double bond. Right? Yes. Right? So, yes, bache. So, lipids with more amount of saturated fatty acid are solid at room temperature. Right? You can relate this point with the cell membrane. Jैसे cell membrane में unsaturation is also there. That is because of unsaturation in the fatty acid, in that hydrophobic fatty acid chain, the double bond will be there. Right? The kink will be there, and it will not let that plasma membrane to get solidify. Okay, so unsaturation is required. So that's how you can relate it with the plasma membrane. Okay, that is how you can relate it with the plasma membrane. So what am I saying here? I am saying that no carbon to carbon double bond, saturated fatty acid, and the lipids with more number of, right, more amount of saturated fatty acid, they are solid at room temperature. Right? Jaise ki your butter, like your ghee, butter and ghee. So obviously when it is the unsaturated, right, they are in liquid at normal temperature. Right? They are liquid at normal temperature. Yes, bache? Yes, bache? Lipids with more amount of unsaturated fatty acids are liquid at room temperature. They are what? They are liquid at room temperature. What are they? They are liquid at room temperature. जैसे कि your mustard oil, right? बच्चे, I think you have seen the advertisement on the TV, right? On the television, where they say na MUFA, PUFA, mono unsaturated fatty acid, poly unsaturated fatty acid means the oils, right? The cooking oils where so many unsaturation is there. There obviously they'll be liquid at the room temperature. Poly unsaturated fatty acid, if that fatty acid is having more than one double bond. Okay, bache? Right, bache? Okay, so mustard oil is there. So remember, when you talk about the animal fat is mostly saturated. Animal fat is mostly saturated, and plant fat is, animal fat is mostly saturated. And if it is saturated, obviously it is responsible for, right? It is basically increases the blood cholesterol, right? Because metabolically it is less active. I'll come to this. And yes, they have high melting point, they have low melting point. So animal fat is mostly saturated and plant fat is mostly unsaturated. It is mostly unsaturated. Okay. Chaliye, so next is कि metabolically they are very less reactive, right? So metabolically less reactive, so they don't take part in part in metabolism and get stored in animal body. And get stored in animal body and result in obesity, right? And they have tendency to store in animal body and result in obesity. Okay, bache? Okay, bache? Yes? Result in obesity. And bache, in liver, then what's going to happen? In liver, these saturated fatty acids will be converted into cholesterol, and you know that if cholesterol will form in our body in excess, right, that will get deposited within our blood vessels, and obviously the blockage can be there. So that's why we say that they increase the blood cholesterol. Your saturated fatty acids increase blood cholesterol, right? They increase blood cholesterol, and now you know the reason why, right? Now you know the reason why. Okay? Okay? Right? Right? So obviously here you can write it now, कि they are metabolically more active, take part in metabolism, so they will not get deposited in our body, they will not cause obesity, they will not result in the cholesterol deposition, and obviously will not increase the blood pressure. Do I need to write it? I think you can write it, right? I think you can write it. Chaliye. So other than that, when you talk about the saturated fatty acids, okay, okay, so here hydrogenation means addition of hydrogen, right? Hydrogenation means addition of hydrogen, right bache? Right? So I'll explain hydrogenation. So basically, bache, when there is the hydrogenation, means when there is the addition of hydrogen, then what's going to happen, right? Let's say if there is a double bond, right? Hydrogen will be added, that double bond will break, then obviously unsaturation will convert into saturation. So it is already saturated. So hydrogenation cause no effect here. On exposure to air, hydrogenation will not occur, and even it will cause no effect. But here, unsaturation is there, double bond is there. So when there is the addition of hydrogen, these unsaturated fatty acids can get converted into the saturated one. It will get converted into the saturated one, right, bache? Right? So in the saturated fatty acids, bache, essential fatty acids are absent, right? Essential fatty acids are absent, and essential fatty acids may be present, or you can say they are present. Now the point here is कि what is the meaning of essential fatty acids? Bache, they are the one that are not synthesized by the body. They are not synthesized by the body. Our body is not capable of synthesizing it. So they are not synthesized by the body. So it should be there in our diet, right? So it should be the part of our diet, right? Part of our diet. Okay, so you have saturated fatty acid, so it is P2S is S2P is the trick. जैसे you have hybridization ना, S2P sp2 hybridization ना, but here you have to keep it S2P. You have to keep it S2P. What is this? S for saturated fatty acid, S for stearic acid, and SP for palmitic acid. P for palmitic acid. So stearic acid and palmitic acid is the example, right? So saturated fatty acids, they are having a general formula. It is CnH2n. Yes, it is CnH2n O2. So O is not N here. it is O2 here. So stearic acid and palmitic acid is there. So your stearic acid mein number of carbon is 18. Number of carbon is 18, total number of carbon. And palmitic acid mein carbon number is 16. Okay, so what is the general formula of saturated fatty acid? CnH2nO2. 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 What is it? CnH2nO2. Right? What is it? It is CnH2nO2. Understood? CnH2nO2. Clear? 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 Okay. So here it's O la la. That's how you have to remember. Right? They are the O la la. So oleic acid, linoleic acid, linolenic acid, and arachidonic acid. So when you talk about the number of double bonds, number of double one is 1, then 2, then 3, then 4. So these 3 come under polyunsaturated fatty acid. You can check that cooking oils, right? You can check that ad, mufa, pufa. These are the words that they use. So mufa mane monounsaturated fatty acid. So monounsaturated fatty acid, polyunsaturated fatty acid. Monounsaturated fatty acid, polyunsaturated fatty acid. Right, bache? Right, bache? And when you talk about the carbon number, here also, carbon number is 18, carbon number is 18, carbon number is 18, but here the carbon number will be 20. So these 3 are having carbon number as 18 and they are having 20. Okay? Right? Right? Okay? Okay, so the general formula here is CnH2n minus 2x, oxygen is 2. So obviously n is the number of carbon here na, but 2x here it will be the double bond. It will be the double bond. So this is for the unsaturated fatty acid. So yes, you need to remember the carbon number as well. This is important. You need to remember the carbon number as well. It is important. Right? It is important. Okay? Okay, so when you talk about the essential fatty acid, right? When you talk about the essential fatty acids, so yes, examples are important. So essential fatty acids में what is the trick? जैसे OLA is the trick for, yes, it is the trick for unsaturated fatty acids ना. So here essential में it's la la la, मतलब linoleic, linolenic, arachidonic. ठीक है? So number of double bond 2, 3, 4. Number of double bond 2, 3, 4. So carbon number 18, 18 but 20. ठीक है? So see, carbon number is 18, oxygen is 2. Carbon number 18, oxygen is 2. Carbon number 20, oxygen is 2. Now H is 2n minus 2x. है ना? H is 2n minus 2x. Now, so H will be here. It will be 32, 30, 32, isn't it? Isn't it? 32, 30 and 32. That's the formula. So these are the essential fatty acids. Okay. These are the essential fatty acids. Okay. Yes. So ये देखो oleic acid, right? So oleic acid carbon number is 18, 1 double bond. ये देखो. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Right? Yes. So see this, your stearic acid, your palmitic acid, 16 and 18. Look at this. है ना? The structure is also easy. The structure is also easy. बाकी linolenic acid, arachidonic acid. So number of double bonds 1, 2, 3, or 4? No, total number of carbon 20. Linolenic, carbon number is 18. Unsaturations 3. That's how it is. Okay, that's how it is. So that's about the lipids. And see this. See this. Look at this. So glycerol. So it's important. Here go, palmitic acid is also given. So yes, 14, 15 and 16. So glycerol is your trihydroxypropane. It is your trihydroxypropane. And this one is your triglyceride, right? So one glycerol plus 3 fatty acids. Okay, and this is your phospholipid. So it's a type of compound lipid or conjugate lipid. So what will you see here? You will see one glycerol. You will see one glycerol. Ye dekho, one fatty acid, second fatty acid, right? So 2 fatty acids plus, see this phosphoric acid H3PO4 plus this colon nitrogenous compound. Nitrogenous compound, write down. So 2 molecules of fatty acid, one is the glycerol, one is H3PO4, one is the nitrogenous compound. So this is your phospholipid. The structure should be clear. So it is the most commonly occurring lipid, and the best example here is lecithin, right? Phospholipid, most abundant phospholipid is your lecithin, right? Lecithin. You remember alveoli of the lungs, they are also covered with the lecithin. It is acting like a surfactant. It is acting like a surfactant, right? It will reduce the surface tension, keep the alveoli open, right? And the exchange of gases will be there. Okay, and the exchange of gases will be there. Okay, okay, right? And the exchange of gases will be there. Okay, Hanji, clear? Clear? So phospholipid, lecithin, you have glycerol will be one only. One glycerol, 2 fatty acids, right? One H3PO4, one nitrogenous compound, colon. Right? Okay. Right? So remember plasma membrane, that's how we draw it. So polar head, non-polar tail, yes, polar head, non-polar tail, that's how it is present. So when it is lecithin, this nitrogenous compound is taken. Right? Yes. So lecithin is also known as your phosphatidyl choline. Okay, so other than that, sphingolipids, cephalin, they all are the examples. Glycolipids are also the conjugate lipids, but obviously we are not going to discuss that in detail. Okay, bache? Okay, bache? So now you can see this diagram here, cholesterol. So it is the most decorated micromolecule in biology, right? It is known as the most decorated micromolecule in biology. You know that they are the water insoluble but soluble in organic solvents, right? They are having that hydrocarbon rings and that tail. So here you guys can see the structure of cholesterol. In the paper it can come. Okay, in the paper it can come. So it is the example of derived lipid, right? Right? Okay. Okay. Chalo. So when you talk about the derived lipids, their structure is obviously more complex, right? So here you see steroids, here you see chromosomes, lipids. Okay, so when you talk about the steroids, you know that they are very important for the formation of sex hormones also. So you have two categories here, sterols and sterones, right? So OH, alcohol is the functional group here. Alcohol is the functional group here, right? Alcoholic steroids they are. Jaise your cholesterol, alcoholic steroid it is, right? And sterone ketonic steroid. So alcoholic steroid, ketone is the functional group here, so ketonic steroid. Ketone is the functional group here, so ketonic steroid. Okay, okay. Just say your sex hormones are made up of these sterones, and this is the cholesterol here. You know the function of cholesterol. Okay, okay. Done? Right? So there is a form of cholesterol is very important. There is a form of cholesterol which is there in our skin. So when exposed to sunlight, it gets converted into the vitamin D. And you know that that's why we say ki, for vitamin D we need to take the sun bath, right? Right. So that's how it works. So vitamin D, that calciferol will be formed from that form of cholesterol. So when it is the sterones, right, they are responsible for the formation of sex hormones, adrenocorticoids, like this. So chromolipid is also a kind of derived lipids. Your terpenes, right, your terpenes, we have discussed it in this chapter only. Terpenes, chromolipids are also known as terpenes, repeated units of isoperines. So this is all about the lipids, right? This is all about the lipids. Okay, so ye dekho. About the lipids, they have explained it. So now the next thing that we need to discuss is amino acid. Okay, so lipids are not the, lipids are what? Lipids are not the biomacromolecules, rather than they are biomicromolecules. So ye dekho, everything regarding lipids is given here in the NCERT. Okay. So do read it from NCERT, and now let's discuss about the amino acids. Wait, I'm looking for something. Let's talk about the amino acids. So ye dekho, about the amino acids also it is given in NCERT, na? It is given in NCERT. So that's what we need to discuss. So do you remember the name of the amino acid? I always flex this thing. Do you remember the name of the, yes, do you remember the name of the amino acids? Right? I mean, name of the amino acids: alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, yes, glutamic acid, alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, GH, histidine, isoleucine, leucine, lysine, methionine, proline, serine, threonine, tryptophan, tyrosine, valine. ठीक है? So magic amino acids they are, building blocks of proteins they are. So they are 10, 11, 12, 13, 14, 15, 16, 17. Okay, I missed few. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine. Oh, okay. Good. Glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine. Yes. Phenylalanine. So 8th, 9th, 10th, 11th, 12th, 13th, 14, 15, 16, 17, 18, 19 and 20. So 20 amino acids, right? So in an alphabetic order, I remember it. And yes, you know that it is even important for the genetic code. So if you can remember, if you are targeting 360 out of 360, then though I'm going to suggest you to remember it. And trust me, it's easy. It will hardly take you 5 minutes. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenyl, alanine, serine, threonine, tyrosine, tryptophan and valine. Okay, these are the 20 amino acids. These are the 20 amino acids, and about the amino acids we have to discuss. But let's have a break. Okay, so in this break time, do revise it. So let's have a break. Let's meet at 11. Chalo, I'll write it here. Let's meet at 11 p.m. Okay, so let's have a break. Done? Cheers. Hello, hello, hello. Hello, hello, hello. What's up? Hello, students. What's up? So, I was checking my old lectures like around 10 months back for NEET 2023. Also, I took the marathon session, right? That too on biomolecules. I was checking that out, and in 5 hours, with breaks, of course, I completed the carbohydrates. I think the lipid part there only. No, carbohydrate or only carbohydrate in 4 hours. But today, see, I have completed the lipids also, and I'm talking about the amino acids. So it's not like that. Yeah, I've missed something. Yeah, I am not discussing everything in detail. Still, I'm discussing the things in detail, but it comes with the experience. And today, I have added many images so that we can save time, right? So that we can save time. So see, I know you people are feeling lazy, and if you are in the northern part of the country, definitely because it's too cold here, so you must be feeling sleepy. Chalo, theek hai yaar, chodo ma'am, we'll discuss it later. We'll watch your previous lectures. Let's end the session. Even I was thinking the same. Even I'm feeling tired, right? And obviously, yes, I'm feeling tired because classes are there in Phoenix batch, in the NEET Avengers fast track batch also. And do you know, in the Phoenix batch, we are about to finish the syllabus. Class 12 syllabus is going on. We are about to finish the syllabus, and here in this particular month, we will complete all our most important units. Okay? And after that, in NEET Avengers batch also, because we have to complete the syllabus till March 31st. So lectures are there every day, plus test is there, so many things are there, right? So that's why, even I'm feeling tired. But still, if you guys support, see, the lesson, the students are very less. It's okay, it's fine. But if you people support, we'll still finish our chapter. What say? We'll still finish our chapter. What say? What say? Let's finish it, yeah. It's just 12:15. Let's wake up till 2, till 3. Let's finish it. कोमल वो कब का हो गया है, दोबारा नहीं करवा सकती, sorry. चलो. So, okay. So now we are going to discuss amino acid. What are we going to discuss now? Now we are going to discuss amino acids. So when you talk about the amino acids, they are bio-micromolecule. Why they are bio-micromolecules? Because obviously their molecular weight is less than 1000 Dalton, right? Because their molecular weight is what? Their molecular weight is less than 1000 Dalton, right? So definitely it will be there in the filtrate, that is in the acid soluble pool. Am I right? It is there in the filtrate, that is in the acid soluble pool. So now when you talk about these amino acids, they are the building blocks. Yes, they are the building blocks of proteins. Means they are the one that will join with the help of peptide bonds, and they are going to form the proteins. Okay, they are the one that will join with the peptide bonds, and they are going to form the protein. So overall, like if I talk about the number of amino acids, so they are around 200 to 300, right? When I talk about the number of amino acids, they are around 200 to 300, right? They are around 200 to 300. But when it comes to the protein forming amino acids, right? When it comes to the protein forming amino acids, so that protein forming amino acids are 20. They are 20. They are 20. So again, I will tell you to remember their name, right? If you are targeting 360 out of 360, right? So if you are targeting 360 out of 360, then so definitely it is important for you. It is important for you to remember the name of the amino acids, and it will be helpful in your MBBS during your course, during your MBBS course also. So it's easy. In the alphabetic order, you can remember alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, MNOP, Q, RS, serine, threonine, tyrosine, tryptophan, and valine. These are the 20 amino acids. These are the 20 amino acids. So they have 3 word symbols also, and they have 1 word symbol also. Just say for an example, A for alanine, R for arginine, like this. And I'm not going to discuss it. I'm not going to discuss the 1 word symbol here. I am expecting you people to do it. Okay, I want you people to do it. Okay, bache? Okay, bache, amino acid and their symbols. Amino acid and their symbols. Okay, one word symbol I'm talking about. So it's easy also. So alanine, arginine, asparagine, right? Okay, so A, R, here it is N, aspartic acid, it should be D. Okay, chalo, I'll discuss it. Then cysteine is for C, glutamin is for Q, glutamic acid is for E, CQE. That is CQE, right? So it is the one word symbol, right? Then HI, L. So lysine is K. So glycine, right? Glycine is G only. Glycine is G only. Histidine is H, right? Then isoleucine is I, leucine is L, lysine is K. And when it is methionine, methionine again will be M, right? Methionine again will be M. Proline P. Phenylalanine I have to check. Phenylalanine, serine will be C. Phenylalanine I have to check. Right, valine is of course V. This tyrosine is W. Tryptophan is T. Okay. Right, so one word symbol may, yes, there are some which even phenylalanine hai to F. It should go like this. Yes, it should go like this. So it is phenylalanine, F. Serine will be F. Okay, tryptophan, sorry, my bad. Tryptophan, my bad. So tyrosine, Y, tyrosine. Tryptophan is W, threonine is T, and then valine. So these are the one word symbols. These are the one word symbols. So alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, right, methionine, proline, phenylalanine, then you have serine, threonine, tyrosine, tryptophan, and valine. So trust me, if you will put efforts, you can remember it. It's easy. And yes, on the channel Unacademy NEET English, I have uploaded a table, right, for the tricks of genetic code. So if you are targeting 360 out of 360, go for it. And it's not the only thing. You have two other amino acids also. 21st and 22nd amino acid, that is your selenocysteine and pyrrolysine. What's that, bache hairy? That is your selenocysteine and pyrrolysine. What's that? That is your selenocysteine and pyrrolysine. What's that? Selenocysteine and pyrrolysine. Okay. It is selenocysteine. And selenocysteine. So its structure is just like cysteine, but yeah, selenium is attached here, and this one is pyrrolysine. So once I was reading about it, so stop codons. Stop codons are there in the genetic code. When we discuss the genetic code, there are 3 stop codons. So sometimes that stop codons, right? That stop codons code for these amino acids. So that is a different discussion, but just for your knowledge, I'm telling you. So these are the 20 amino acid, and these are the one word symbols: alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, and all. Okay. So 3 word symbol: alanine, AR, ALA, arginine, ARG, asparagine, ASN, etc. It is also like this. Okay, so they are the building blocks of the protein. They will join with the peptide bond. They will form the protein. And as I said, protein forming amino acids are 20 as per your NCERT, but it is 22. Selenocysteine and pyrrolysine is also there. So overall in proteins, carbon, hydrogen, oxygen, nitrogen is there, and sulfur is also there. Sulfur is also there in some amino acid. So again, it is important. So here we have a trick, that is CMS. What is CMS? CMS means Chief Minister Sir. So what is this? It means that cysteine and methionine. C stands for cysteine, M stands for methionine, S stands for sulfur. So cysteine and methionine, they are sulfur containing amino acids. This is important. They are sulfur containing amino acids, right, bache? So some may have sulfur, okay? Right, bache? Okay, so what are these amino acids? So amino acids, definitely they are organic compounds. What are they? These amino acids, they are organic compounds indeed, right? Indeed. What are they, students? They are organic compounds indeed. And these are the organic compounds having amino group and carboxylic group, right? It is having amino group and carboxylic group, and moreover, they are the substituted methanes. They are the substituted methanes. What are they? They are the substituted methanes. So it is CH4. Methane is your CH4. What is methane? Methane is your CH4. Yes, Anuradha. Methane is your methane is your, yes. So it is CH4. So when you talk about this now, see, substituted methane. So let me explain this. So methane is CH4. Now see, 4 bonds are here. So here you will see amino group NH2. Here you will see carboxylic group, acidic group. One side will have hydrogen and another side, either it can have H or R. It can have H or R. See, this is your carbon. Here you have amino group, the basic group of course. Here you have carboxylic group. This side is having hydrogen and this side either H or R. Either H or R. Now. If it is like this, look at this. It is glycine. It is glycine. And this glycine is the simplest amino acid. This glycine is the simplest amino acid. What's that? This glycine is the simplest amino acid. So here, right, because this side is also having hydrogen, this side is also having hydrogen. This is the simplest amino acid. Now, let's say if it is like this, NH2 group should be on the left side, COH group, because generally L amino acids are there in our body, right? So NH2 group is on the left side. So here if you have hydrogen and at the place of R, if you have CH3, then this is your alanine. This amino acid is your alanine. So now when you look at this carbon, all the 4 sides are having different amino acids, different group. All the 4 sides are having different group, right? All the 4 sides are having different groups. So this is your asymmetric carbon, alpha carbon. So that's why amino acids are also known as alpha amino acids. They are also known as alpha amino acids. So this is the alpha carbon because all the 4 sides are having different functional group. But that is not the case in the case of your, glycine. Okay. So in L amino acids, NH2 group is on the left side and mainly we are having the L amino acids. Okay. NH2 group is on the left side. Understood? It is on the left side. Understood? Yes? Yes? Done? So बच्चे, except glycine, all other amino acids are optically active. All other amino acids are optically active. And this R group will tell us about the nature. It will tell us about the nature of your amino acid. Okay, so they are substituted methanes. Right, bache? And moreover, amino acids are what? They are amphoteric compounds. Right, they are amphoteric compounds. Why? Why do we call it as amphoteric compounds? Because they have both NH2 and COOH group. Acidic and basic group is there. That's why it is known as amphoteric, right? That's why it is known as amphoteric, right, bache? Right, bache? So alkaline and acidic, both the groups are there, right? Now, bache, at isoelectronic point. At what? Now, the next thing that we need to understand here is Zwitter ion. So, at isoelectric point, amino acid behaves like a Zwitter ion. Amino acid behaves like a Zwitter ion. It behaves like what? It behaves like Zwitter ion. Is that clear? Is that clear? Yes, at isoelectric point, they are present in the form of Zwitter ion. Okay, bache. So basically, I told you that, right? It behaves like Zwitter ion. So see, in the case of amino acid, because NH2 group is there, right? NH2 group is there and COOH group is there. So can I say that they are ionizable? Means amino acids can form the ions. Amino acid can form the ion. It is ionizable in nature. It is having the NH2 group. It is having the COOH group. So I am saying that it is ionizable in nature, right? It is ionizable in nature, right, bache? Right? Because of this NH2 and COOH group, it is ionizable. It can form the ion. In the solution of different pH, it can form the ions, and the structure of this amino acid will change. And the structure of this amino acid will change, and they are amphoteric. Why? Because they have NH2 group as well as COOH group. So I am saying at isoelectric point, amino acid is a Zwitter ion. So what is the meaning of Zwitter ion? See, I can write, okay, CHR NH2 COOH. Now see, NH2 is there, so it can become NH3 positive. So it is basically what? It is the cation, positively charged cation. Now this one, it's COO negative. It is anion. Then it is what? Then it is anion. Obviously, in the solution of different pH, it is going to behave like that. Now when it is the Zwitter ion, so it will be like this, NH3 positive and COO negative. So here in this particular compound, the positive and the negative charge will be there. See this, NH3 positive, COH negative. This H will go here. NH3 positive, COH negative. So it is your Zwitter ion. It is your Zwitter ion, right? Also known as dipolar ion, right? Because two charges are there, but one is plus, one is minus. So it is electrically neutral, right? 1 plus 1 minus is there. So it is electrically neutral. Is that clear? 1 plus 1 minus is there. So it is electrically neutral. Understood? Understood? Okay. Right? So it is electrically what? It is electrically neutral. So this is the Zwitter ion form. So this is important, even given in your NCERT. You can see this. Ye dekho. That's the Zwitter ion. It is amphoteric in nature. Okay. It is amphoteric in nature. Okay. Right? Chalo. So now write down the important information. Write down the important information here. See, I told you it is the R group that is going to decide what type of amino acid is there. So when it is acidic amino acid means it will have more COH than NH2. It is having more COH group than NH2 group. Right. So when you talk about the acidic amino acid, see, glutamic acid and aspartic acid, simple. Glutamic acid and aspartic acid, simple. Yes, glutamic acid and aspartic acid, simple. Yes or no? Yes or no? Just for an example, see, NH2 COOH. Here you have hydrogen. Here you have Right. So glutamic acid and aspartic acid, because it will have more COH. So how can you see this? So R group will be CH2, CH2, and here you will have COH. So basically 2 COH group, 1 NH2 group. Glutamic acid, because net charge will be, right, 2 negative, 1 positive. So net charge is going to be negative here. So what is it? It is the acidic amino acid, glutamic acid, right? It is the glutamic acid. What's that? It is the glutamic acid. Understood? Understood? Yes? Understood? Sure. So aspartic acid, its structure is very simple. Its structure is very simple. Here they go. C, H, NH2, COOH. Here you have 2 CH2. Here you will have only 1 CH2 and 1, 2, 3, 4. C double bond O, OH, like this. So that is your aspartic acid. So these two are the acidic amino acids. These two are the acidic amino acids, yes or no? These two are the acidic amino acids, yes or no? Yes or no? Right? Now when we talk about the basic amino acid, right? For basic amino acid, so basic amino acid means positively charged. Basic amino acids means, bache, they are positively charged. Basic amino acid means they are positively charged, and here you have trick, that is Hall. Here you have trick, that is Hall. HAL. HAL. So H stands for histidine, A stands for arginine, and L stands for lysine. So what is the trick here? Histidine, arginine, lysine, Hall. Histidine, arginine, lysine, Hall. Histidine, arginine, lysine. Okay, so here you will see there will be 2 NH2 group and 1 COOH group. So net charge will be positive. So histidine, arginine, lysine, Hall is the trick. Histidine, arginine, lysine. And remember, yes, histone proteins, do you remember? Histone proteins. So histone proteins are positively charged. Our DNA will coil over it and then will form a nucleosome, the chromatin, remember? So these histones are positively charged, so they are having lysine and arginine. So it is a repeated NEET PYQ in 2020. It was there, I think in 2022 or 1, it was all, again, it was repeated, right? About the histone. So they are loaded with lysine and arginine. So sometimes some students think only lysine and arginine, only these two amino acids are basic. No, even your histidine is the basic amino acid. So these 5 acidic and basic, remaining 15 are electrically neutral. Remaining 15 are electrically neutral, right? Remaining 15 are electrically neutral. Understood? Understood? Remaining 15 are electrically neutral. Now the next point, I said glycine is the simplest amino acid. Tryptophan is the most complex amino acid. It is the most complex amino acid. So tryptophan, you remember, for auxin formation it is required, right? Auxin is a plant growth regulator. It is a PGR. So for its formation, your tryptophan and zinc ion is required, right? Your zinc ion and tryptophan is required. This is important for the tryptophan, right? This is important for the tryptophan. And even the, right, it is advised to drink milk before sleeping, right? I hope you know that it is always advised to take milk before sleeping. Why? Because that milk is having tryptophan. That tryptophan, right, this amino acid helps in the synthesis of what? It helps in the synthesis of something. It helps in the synthesis of dash, and that dash induces the sleep, right? Tryptophan helps in the synthesis of dash, and that dash induces the sleep. Anybody in the class who can answer it? I've repeated it many times in my lectures. Yes. Yes. Anybody? Anybody? Yes, anybody? No. बच्चे tryptophan, firstly I told you that it is important for the synthesis of your plant growth regulator auxin, and zinc ion is also required for that. Secondly, I am saying that it is generally advised to take milk before sleeping because it is said that milk induces the sleep. What does it mean? See, tryptophan, it helps in the synthesis of serotonin. You know, serotonin, also considered as happy hormone, neurotransmitter, serotonin. So that serotonin then get converted into melatonin. I'm not discussing the reaction here. I'm simply saying that this tryptophan forms serotonin, and then it will further be converted to melatonin, right? Melatonin, pineal body release melatonin. That also induces the sleep. That's how it works. Okay. That's how it works. So it is for the tryptophan. Now, another amino, so it is the most complex amino acid, tyrosine. And yes, it is the aromatic amino acids, right? It is the aromatic amino acid. So even your tyrosine, even your tryptophan, and even phenylalanine. This is also aromatic amino acid. The ring is there in its structure. This is also aromatic amino acid. So tyrosine helps in the melanin formation. So melanin is responsible for our skin pigmentation, right? So for melanin, we need tyrosine. Okay, and not just melanin, even the adrenaline, noradrenaline, thyroxine, even adrenaline, noradrenaline, and even for thyroxine formation, tyrosine is required, right? For this, adrenaline is your, remember epinephrine, right? Epinephrine, yes, right? These are the emergency hormones, na? Epinephrine, norepinephrine, adrenaline, noradrenaline, same, na? And thyroxine, it also needs tyrosine. That's how the amino acids are important. And I don't know whether you know it or not. Other than that, even phenylalanine, right? Phenylalanine is also the aromatic amino acid. So these three amino acids are what? They are the aromatic amino acids, right? These three amino acids, what are they? They are aromatic amino acids. These three amino acids, what are they? They are aromatic amino acids, right? They are aromatic amino acids. Okay, right? Pakka? Done? And in class 12, in genetics, you remember phenylalanine hydroxylase, there is a disorder, right? Out of nowhere I'm discussing it. Phenylketonuria. Anybody in the class? Phenylketonuria. Remember autosomal recessive disorder? Yes, that's an autosomal recessive disorder. Right, so here in this disorder, phenylalanine hydroxylase is not forming. There is a mutation and we are not getting this particular enzyme. Right, there is a mutation and we are not getting this particular enzyme. Because of that, what is happening? Actually, bache, this enzyme convert phenylalanine hydroxylase. This enzyme convert your phenylalanine into tyrosine, and that tyrosine, as I said, is important for melanin formation. So if this enzyme is not there, your phenylalanine, it will be accumulated, right? So tyrosine will not form, so skin pigment issue will be there, plus mental retardation will be there, right? So it can cause this. So we discuss it in class 12, the genetics, phenylketonuria. So here we are talking about the amino acids, so I thought that I should discuss the same with you all, right? You can note down it. Okay, you can note down this. Okay, yes, okay. Chaliye. So now the next thing is essential amino acid. So I hope now you know what is the meaning of essential amino acid. Then I will discuss the peptide formation. So ye dekho. So these three structures are given in NCERT. Do discuss it. Glycine, alanine. So if it is H, glycine, CH3, alanine. If CH2OH, then it will be serine. So that's important. And already we have discussed glucose and ribose. So it is the alpha or beta. This one is alpha ribose. In the case of beta, hydrogen will be down. ठीक है, हो गया? Now, Okay, so amino acids done. So a basic structure of amino acid done. Here they go NH3 COH group, and here you people can see, right? 20 amino acids, side chains, charge at physiological pH, positive, negative, arginine R, histidine H. So these three are basic amino acids. Okay, these three are basic amino acids, right? So acidic amino acids also discussed, and as I said, selenocysteine, 21st amino acid. So rest structure is just like your cysteine only, but here at the place of SH, right? It is the sulfur containing amino acid now. You will see selenium is there. Okay, and in the case of proline, proline is not an aromatic amino acid. It is at the place of NH2 group, it is having NH. That's the special about proline. Baki, they all are electrically neutral. So your phenylalanine, tyrosine, tryptophan. Phenylalanine, tyrosine, tryptophan. Phenylalanine, tyrosine, tryptophan. Right? They are the aromatic amino acids. They are the aromatic amino acids. Done. Okay? Okay. Okay. So, yes. Next is your essential amino acids. See, that is important. So if you remember the name of essential amino acids, whatever is left, keep it in non-essential category. So essential amino acids are the one, they are not synthesized. They are not synthesized by our body. So they should be the part of our diet. So they should be the part of our diet. You're getting my point? So they should be the part of our diet. Is that clear? Is that clear? Is that clear? So what is the trick here? The trick here is private. For private, we write it like this, PVT. Right? Private, short form. Tim. Private Tim Hall. Private Tim Hall. What's the trick? Private Tim Hall. So P stands for your phenylalanine. P stands for your phenylalanine. V stands for, of course, valine. Right? T stands for threonine. Right? This T stands for tryptophan. This I stands for isoleucine, of course. M stands for methionine. M stands for methionine. H stands for histidine. And see this trick, here in this trick, this is just a trick. We are not using one word symbol. Hello, hello, hello. Right? This is just a trick. We are not using one word symbol here. Right? One word symbol is not there. It is just a trick, private Tim Hall. Private Tim Hall. So A stands for arginine, L stands for leucine, and L stands for lysine. So these are the essential amino acids. Essential amino acids. So in some books it is mentioned that histidine and arginine, they are considered as semi-essential. They are considered as semi-essential. Why? Why? Because they are synthesized in the adult body. Right? They are considered as semi-essential. Why? Because it is believed that they are, yes, they are semi-dispensable. They can be synthesized in adults. Right? They can be synthesized in adults. Okay? Done? Yes. Okay. So now, you know that these amino acids, they will join, and they will form proteins. So when we talk about the proteins, they are the biomacromolecules. So because proteins are biomacromolecules, definitely they will be acid insoluble fraction. Right? Acid insoluble fraction. Okay, bache. Right? So they are responsible for the basic framework of our body, right? Proteins, you know that mostly enzymes are proteins, so they are very useful for our biological activities. So when you talk about the proteins, amino acids, I told you already, they form proteins, okay? And when you talk about the proteins, remember one thing, they are always, see, these amino acids, they will join together, and they are going to form the proteins, right? They are going to form the proteins. So these amino acids, they will join together with the help of a bond. That bond is known as peptide. So many amino acids, they will join, right? And they will form these polypeptides. Poly means many, polypeptides, and many polypeptides join to form the protein. So in the case of proteins, the one thing that you always need to remember is they are heteropolymers, right? Like in the carbohydrates, we were saying that there are some polysaccharides where same type of monosaccharides are there, right? But these are the proteins, and these proteins are what? They are the heteropolymers. These proteins are what? They are the heteropolymers. Means they are made up of different amino acids. Same type of amino acids are not making it. They are made up of different amino acids. That's what you need to remember. So now let's understand what is this peptide bond. So bache, when we say peptide bond, it's CONH bond. Remember it like this. It's CONH bond. Remember it like this. CONH. I'll show you. You have one amino acid. L amino acids are common now. You have first amino acid. Now you have, this is your second amino acid. Chalo, I'm writing NH2 like this. Okay, so basically again, what will happen here? Minus H2O. Water will come out. And when water will come out, this is the first amino acid. This is your first amino acid. This is your second amino acid. So CO, carboxylic group of first amino acid, will form bond with the amino group of next amino acid. So water will release, right? And then it will join like this. So that's why we say it is CONH bond. C double bond ONH. Right? C double bond ONH. So when two amino acids, when they join together, we call it as dipeptide. What do we call it, guys? We call it as dipeptide. Understood? Done? Done? So it is the dipeptide. What is it? It is the dipeptide. Understood? It is the dipeptide. So that is how many amino acids will join. Just let's say if this is the first amino acid, obviously amino group is not involved in the bond formation. So this will be the N-terminal. And when you will see the last amino acid, the carboxylic group will not be there in the bond formation. So this will be the C-terminal. So in the polypeptide, you will see N-terminal, you will see C-terminal. You will see N-terminal, you will see C-terminal. Is that clear? Yes? Any doubt? So pure protein is normally going to have these two ends. Left end is your N-terminal, last end is your C-terminal. Okay? So starting is the amino terminal, last one is the carboxylic terminal. Okay? This in the NCERT it is given now when we talk about the structure of protein, that in inorganic chemistry you go for the formula, right? Then in organic chemistry you go for the 2D view. Physicists, they describe the 3D view. And in biology, when we discuss the protein, we go for the yes, quaternary structures, yes or no? We go for what? We go for the protein structure in 4 levels, right? And that's what we need to discuss now. That's what we need to discuss now, right? So see, this information done. Okay, this information is already discussed here. Okay, so before starting the structure of protein, I'm telling you this table is also important. So when it is the collagen, you know that it is the most abundant protein of animal world. It is the most abundant protein. What's that? It is the most abundant protein of animal world, collagen, right? And we also write it like this. It is the most abundant protein of extracellular matrix, right? That's why we say it is the most abundant protein of animal world. So you can say it is the most abundant protein of animal world, yeah, extracellular matrix. Then rubisco, remember rubisco, ribulose, bisphosphate, carboxylase, oxygenase. So it is the most abundant, protein of biosphere. And do check it, most abundant organic compound on earth. Do check it, right? Which one is the most abundant organic compound on earth? Do check it. Now look at this table, students. Collagen, intercellular ground substance, trypsin. It is an enzyme. Remember, it's a simple protein, trypsin. It is an enzyme. You should know about it. It's a simple protein, right? Only protein is making it. Trypsin. So trypsin, obviously, it is present in the pancreatic juices. It is present in the pancreatic juices, bache. And this trypsin is released in inactive form. That is zymogen. So when any enzyme is released in its inactive form, we use the word zymogen. When any enzyme is released in its inactive form, we use the word zymogen. So zymogen means inactive form of the enzyme, and here it will be trypsinogen. Okay. So insulin is a protein hormone, right? It's a protein hormone having 51 amino acid in its active form. So insulin is also released in its inactive form. We call it as proinsulin, having 3 chains A, B, C. So in the active insulin, A and B polypeptide chain is there, having total 51 amino acid. Baki antibodies, they are also the protein. They fight with the infection. Receptors present on cell surface, right? They help us to help our immune cells to detect which one are the body cells, which one are not the body cells. Okay. Right. So what's the word here? Receptor is the word here. Right. GLUT4, very important. Right. We have discussed the structure of the plasma membrane. We know that proteins are present. We know that channel proteins are there, carrier proteins are there that help in the transport of the things. Right. That help in the transport of the things. Okay. So GLUT4, that's a PYQ. Right. That's a PYQ. So GLUT4 enables the glucose to transport into the cell. So for the entry of glucose, transporters, these carrier proteins, these channel proteins are required. So GLUT4 is one of them. Right? GLUT4 is one of them. Okay. So you should know this. Now we are talking about the structure of amino acid. As I said, primary, secondary, tertiary and quaternary structure will be there. Primary, secondary, tertiary, quaternary structure will be there. When it is the primary, simple. All amino acids in a linear chain, in a straight chain, they are present. So this, because proteins, they also behave like enzymes. Proteins, they are also behaving like receptors. They are also the channel protein. So obviously, right, when it is the straight chain structure, amino acids are added, right? Amino acids are joined. They are joined, obviously they are connected with the help of peptide bonds. So that primary structure, that linear chain, it's a very simple structure. It's a linear chain. It gives specificity to that particular protein. So here you people can see when it is the primary structure, When it is the primary structure, the sequence of amino acid, that is the positional information in a protein, which is the first amino acid, which is the second, and so on, is given. So primary means it's a linear chain. The sequence of amino acid is clear, right? That which amino acid is present where, like this, and it is imagined as a line. Left hand, of course, represents first amino acid, the N terminal. Right end or the last end will be your C terminal, of course. It will be your C terminal, of course. So protein, a protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of, okay, so that's it. So this is about the primary structure of the protein, right? The linear chain. This is about the primary structure of the protein, linear chain, right, bache? Right, bache? So obviously you will see the peptide bond, right? So primary structure describe type, number, sequence, and order of amino acid. Peptide bonds will be there, right? In the primary structure, peptide bond will be there. But if sulfur containing amino acids are there, then disulfide bond can also be there. So cysteine is going to form that. Cysteine is going to form that. Okay, cysteine is going to form that. Okay, so the specificity of the protein is on the basis of its primary structure. The specificity of the protein is on the basis of its primary structure. So remember, newly formed proteins are always in their primary structure, then further folding will be done as per the need. So primary structure, it is the most unstable one. It is the most unstable one. Got it? It is the most unstable. So newly formed proteins on ribosomes have primary structure. So not just the peptide bond, disulfide bond can also be there. Disulfide bond can also be there. Then comes the secondary structure. Now primary structure was just a linear chain. Now it is secondary. Some coilings will be there. So here you talk about the alpha helix and beta pleated structure. You talk about the alpha helix and beta pleated structure. What you discuss here? Alpha helix and beta pleated structure. Alpha helix, beta pleated structure. Is that clear? Is that clear? Is that clear? So when it is alpha helix, it is right-handed. Remember this. This is important information. It is right-handed. Okay? It is right-handed. What the structure is something like this. Right-handed. Okay, so it is right-handed, right? Right-handed protein. So basically it is the spirally coiled chain, right? Spirally coiled chain. What will be there? Spirally coiled chain. So folding and coiling will be there. Folding and coiling will be there. So when it is the alpha helix, right? So it is right-handed. It is right-handed. It is in the form of a regular spiral or in the form of helix, right? You can say that in the form of a regular spiral or helix. So it's just like the revolving staircase. It is just like the revolving staircase. Is that clear? Is that clear? So when you talk about this right-handed structure, alpha helical structure, what will you see here in the helical structure? What will you see here in this alpha helical structure? What will you see here in this alpha helical structure, my dear students? You are going to see. What will you see here in this alpha helical structure? You people will see intra means within the molecule, intramolecular hydrogen bonding. What you people are going to see here? You will see intramolecular hydrogen bonding. What will you see here? You will see intramolecular hydrogen bonding. Understood? Understood? Intramolecular hydrogen bonding. Yes, bache? Yes, bache? And that will be in between the oxygen of first amino acid. Oxygen of first amino acid and. So what is there? What is there? Oxygen of first amino acid residue and NH group of next fourth amino acid. NH group of fourth amino acid. So intramolecular hydrogen bonding will be there, right bache? So the proteins, just like your tropomyosin is the example here, keratin is the example here, myosin is the example here. So these are the examples of alpha helical structure: keratin, myosin, tropomyosin. Right bache? Right bache? Okay, so next is your beta pleated structure. So when it is the beta pleated structure, my dear students, here one or more polypeptide chains, right? One or more polypeptide chain, they join with the help of intermolecular hydrogen bonding. Here it is intra, here it is inter. Intra mane within the molecule, hydrogen bonding in between the molecule. So either you can have one or two or more polypeptide chains that are connected, and sometimes one polypeptide chain is basically in a zigzag manner, right? It is folded. It will form a zigzag manner. Just say we have the pleated structures, no, pleats of skirts like this. So that protein chain will be folded, intermolecular hydrogen bonding will be there, and you will get the beta pleated structure, right? So intermolecular hydrogen bonding, and they may bend into parallel folds to form the pleated structure. Is that clear? So here you have the example, and that is the fibroin of silk, right? So when you talk about the examples here, what do you have? You have the, yes, you have the fibroin of silk. Okay, yes, fibroin of silk, right? Fibroin that is present in silk. Now keratin, I hope you know keratin. Just are here these days, so keratin treatment, right? Shampoos are loaded with keratin. It's a protein, right? Our dead parts, our nails, are here, they have the keratin protein. So this keratin protein is having abundant cysteine amino acid. So cysteine amino acid is too much here. Cysteine amino acid is too much here. Is that clear? Is that clear? Yes. Is that clear? Next is bache tertiary structure. See this. Now, other regions of the protein thread are folded into other forms. That is a secondary. We have discussed it. Now, sometimes long protein chain, it is also folded upon itself like a hollow woolen ball, right? Sometimes they are more folded. They are more folded, and then they are forming the tertiary structure, right? So this tertiary structure, bache, it gives us a 3D view, three-dimensional view. And when the proteins are in their tertiary structure, they are biologically active. They are biologically active. So when they are in their tertiary structure, they are biologically active. When they are in their tertiary structure, they are what? They are biologically active. Biologically active. So tertiary structure, it is written here na, is absolutely necessary for the many biological activities of protein. It is absolutely necessary for the many biological activities of the protein. Tertiary structure. So enzymes, right? Mostly, most of the enzymes are proteins. So when they are proteins, they are basic. When they are enzymes, it is in their tertiary structure. It is in their tertiary structure, right, bache? How are they present? They are present in their tertiary structure. So look at this diagram. So when you talk about the tertiary structure now, obviously here also you will see the bonding. Indeed. So one thing is for sure. One thing is for sure, that peptide bond will be there. Peptide bond will be there. But other than peptide bond, right? So peptide bond will be the strongest bond in protein for sure. It is the strongest bond in protein for sure. For sure. So other than the peptide bond, disulfide bond is also there. Disulfide bond is also there. Right, bache? Disulfide bond is also there, and this bond is the second strongest covalent bond, and it stabilizes the right. So it is the second strongest bond, bache. It is the second strongest bond, bache, and it stabilizes the, yes or no, it stabilizes the, when you are talking about the tertiary structure, hydrogen bond, ionic bond, even the hydrophobic interactions, everything will be there, right? Everything will be there. So when proteins, they are in tertiary structure, they are biologically active. So tertiary structure mein the further coiling is there na. So we see that, you know, they are coiled and they will form kind of sites here. You remember? Right? And we call them as crevices. Right? And these crevices are nothing. These are the active sites where the substrate will fit in. Okay? Where the substrate will fit in. Where what is going to happen? Where the substrate will fit in, an enzyme substrate complex will form, and obviously then the transition state, because making and breaking of bond will be there, and finally you people are going to get the protein. So these pockets will form the active sites. Okay, right? So next is your quaternary structure. So bache, sometimes, right, this is quaternary structure. We have the example of hemoglobin. Right, let me tell you, hemoglobin, a respiratory pigment, is the example of quaternary protein, and your myoglobin, right, the pigment in the muscles, right, oxygen storing pigment in the muscles, it is the tertiary structure. So quaternary mein 2 or more polypeptide chains of tertiary structure, they unite by different types of bond to form quaternary structure. Right, bache? Yes, to form quaternary structure. So basically the proteins which are coiled in their tertiary forms, that they will 2 or more than many polypeptide chains, they'll come together with the help of bonds, they'll join, they'll form the quaternary structure. So different types of protein chains can make it. So we have, let's say hemoglobin. You already know this. In the case of hemoglobin, we have 2 alpha chains and we have 2 beta chains. We have 2 alpha chains and we have 2 beta chains. 2 alpha, 2 beta. So basically these chains are not similar. They all are not same. So it is dissimilar type. Of chains that are making this quaternary protein. Insulin, in the active form of insulin, you have A chain and B chain. They are also not same. Right. So you have similar type of, yes, you have similar type of chain and dissimilar are also there. So dissimilar mein hemoglobin and insulin is a very common example, or similar mein lactate dehydrogenase. Right? In the lactate dehydrogenase, all the protein chains, all the peptide chains, they are same. They are of same time. Lactate dehydrogenase. ठीक है जी? Okay. So some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides are arranged with respect to each other, it can be linear string of spheres. Yes, spheres are arranged over one another, right? So that will basically your quaternary structure, right? So hemoglobin is having 4 subunits, 2 alpha, 2 beta. So here dissimilar chains are there. Lactate dehydrogenase is having same type of polypeptide chain. So that's what you need to remember. That's it. That's all. So next part here is denaturation of protein. Yes. Next part here is denaturation of proteins. Anybody in the class who can explain this? Very common it is, right? Yes. देखो बच्चे, the changes in pH, the changes in temperature, all that changes can affect the protein structures, right बच्चे? So change in pH, change in temperature, even in pressure, sometimes even the salts, if they get changed, there will be the changes in the protein structure. So protein are found in biological system with 3D view, with 3D structure, right? मतलब proteins when they are present in their 3D structure, in their 3-dimensional view, right? So they are biologically active, and we even call them as native protein, right? They are biologically active, and we call it as native protein. What do we call it, guys? What do we call it? We call it as native protein, right? Native protein. So when it is subjected to any kind of change, let's say temperature is getting changed, pH is changed, pressure is getting changed, metal ions are getting changed. So basically what's going to happen? The hydrogen bonds will be disrupted. H bonds will get disrupted. Yes, hydrogen bonds will get disrupted. So do you think that protein structure can stay in its 3D form? Do you think that this protein structure can stay in its 3D form? Of course not. Of course not. Of course not. Yes or no? Yes or no? Yes or no? Yes? Yes? Right. Just say, let's say if it is the tertiary structure, it will be something like this. So bonding is getting disrupted, it will become helical, and then basically it will come to its linear form now. So if it is a tertiary structure, if there are such changes, then protein can change into its secondary form, then it can change into its primary form. But I told you biologically it is active in its 3D form, right? It is active in its 3D form, right? So this is the denaturation of protein, in which tertiary structures will destroyed, will form primary, secondary and tertiary will destroyed and will form primary, right? But primary structure will remain intact. Okay, so when there is the denaturation of proteins, the changes are there in temperature and pH that can result in the disruption of these hydrogen bond. So tertiary structure can convert into secondary, then to primary, and secondary can convert into primary. So primary structure will remain intact. Please understand this. Primary structure will remain intact even during the denaturation. So primary structure, it remains intact. It remains intact. Primary structure, it remains intact if it is the denaturation of protein. Okay, so we have the examples. We have the examples, right? Just say your coagulation of egg white. You know na? Egg white. Which protein is it? Albumin. MEN it is. MEN. It is not MIN. Albumin is in our plasma. It is albumin. MEN. So when we heat that egg white, right, this albumin, it gets coagulated. That is also the denaturation of protein. Curdling of milk. Curdling of milk. Yes. Curdling of milk. Milk curdling. Coagulation of milk. That is also the example of denaturation. So you know that when the lactic acid bacteria, because when you talk about the milk, I think I forgot to tell you that lactose is the milk sugar. Lactose is the milk sugar. Lactose is what? Lactose is the milk sugar. So in the milk, the sugar is lactose and protein is casein. Casein. The protein is casein and the sugar is lactose. The sugar is lactose and the protein is casein. Casein. So lactic acid bacteria, it acts on this milk sugar lactose. Right, lactic acid bacteria, while formation of curd, this is going to happen now. So lactic acid bacteria, lactobacillus, it will act on milk sugar lactose. It will do the fermentation. Bacterial fermentation will be there and lactose will get converted to lactic acid. Now this lactic acid acts on milk protein casein, and will coagulate it, right? That will result in the formation of curd. That is the curdling of milk. That is also the example of denaturation here. That is also the example of denaturation here. Is that clear? Is that clear? Sure. So this is all about the proteins. Right? So bache, there is one Ramachandran plot. There is one Ramachandran plot, right? I'm not saying you have to discuss about it in detail. It is related to proteins, right? It is related to protein. So Ramachandran plot is a graph which helps to analyze allowed conformations of protein. It helps to analyze allowed conformations of proteins. How is it going to analyze? It is going to analyze the allowed conformation of proteins. Is that clear? And it is the most outstanding contribution in structural biology. So if Ramachandran plot is there, it is associated with protein. It helps to analyze the allowed conformation of that proteins. Is that clear? Is that clear? Yes. Yes. Right. So you know that the role of protein, right? That collagen, that all, they are used in hydrolyzed collagen, elastins, they are used in skincare products, keratin is used in hair care products. So I hope this part is clear to you all. Yes, I hope this part is clear to you all. Right? Okay, so proteins are having different roles. And do you know on the basis of, they are also having fibrous structure and globular. If it is straight chain, threaded form fibrous, if it is globular, right? So on the basis of that also we divide the proteins. Okay, on the basis of that also we divide the protein, fibrous and globular. But okay, let's not dive deep into that. Okay, so fibrous are thread-like long and rounded, folded will be your globular. Okay, that's it. Dekho bache. Now we are left with nucleic acid and enzymes. Okay, now we are left with nucleic acids and enzymes, because as per your syllabus, that metabolism, all that things are deleted. You have to discuss the enzymes and the nucleic acid. So when you are talking about the nucleic acids, they are what? They are also in the acid insoluble fraction. They are also in the acid insoluble fraction. And you know it very well that it is also a polymer. And it's a polymer of nucleotides. It's a polymer of nucleotides. Right, bache? And or we also call it as, it's a polynucleotide chains are making this. So when I'm saying nucleotide, it is having three things: nitrogenous base, sugar, and phosphoric acid. Be specific about it. Phosphoric acid. Right? So about the nucleic acids now, we will discuss about the nucleic acid. We will discuss in the chapter molecular basis of inheritance. Right? About the nucleic acids, we will discuss in the chapter, right, molecular basis of inheritance. Right, bache? In the molecular basis of inheritance, we will discuss about the nucleic acid in detail. Secondly, enzymes. Most of the enzymes are protein. So, bache, for enzyme, because it will take another 2 or 2.5 hours if we have to discuss it in detail, and this is one of the most important topic. So, I will put a recorded video for that. Right? Even I have already completed it on the channel. You can check it, but I will put a recorded video of it on 30th December or 31st December. Okay? Then, right, and in that particular video, only enzymes will be discussed in a crisp way. Right? So today we will complete it up to this part only. You make sure you read the NCERT. I have added the screenshots from the old NCERT, so you guys can see, you people can understand this bonding in a better way. Okay? So do revise it, do solve the question. So enzyme part, for I'll make a short video, and in that I'll discuss everything related to the enzymes, about the sites and about everything. Okay? So today we will complete it up to this part only. I'll share the PDF in our Telegram group. Okay? And do not forget to join the batch, guys. Do not forget to join the batch. Okay? And in this batch, right, no doubt, we are telling you that we will complete the syllabus till 31st of March. And yes, we are going to do it, because in the month of April, you have to do the self-study and the revision is important. But in this batch still, bache, we have started in the biology, we have started with the most important chapters. And that chapters will definitely ensure your 200 plus marks. So I am planning to complete that most important chapters, right, till January. Right? So till January, if you will complete the most important part of the biology, then obviously in the month of February, you can revise it again and again. And in the February, we'll go for the not so important chapter. So in this way, you can complete your syllabus in a better way. Right? So without wasting time, be the part of this batch. So far, we have just completed one chapter, and today, that is Sunday, right, you are having your first test. So from Monday, cell cycle and cell division will start. Okay? Okay? Right? cell cycle and cell division will start. Biological classification is about to start. So next week, diversity, cell cycle and cell division, then genetics part is about to start. So we will complete the most important units first. So without any delay, be the part of the batch. Okay, so in the description box, you know that the link is there. Baki, if you like the class, do let me know in the comment section, and for the enzymes, I'll definitely put the video. Many students, they ask me, what about the chapter neural control and coordination? It is not available on the channel. Don't worry about that. You know that Monday, see, this neural control and coordination will be discussed on Monday, right? Monday, we will complete this chapter. Okay, so we'll share the notes in the Telegram group. Okay, in our official Telegram group, I'll share the PDF, right? Do revise it, bache. Do revise it. Okay, and stay tuned to our channel. So if you're new, do subscribe the channel. If you're old, be consistent, be consistent and make sure you revise it. So thank you so much. Take care. Bye-bye.